a very, very successful 2 days of conferencing style, 5 days of the entire workshop. And this will be the primer for the rest of the other 3 workshops, which we're expecting to launch this year. In these workshops, we have been able to train early career scientists in the areas of genomics and bioinformatics. And the way we do it is that we have 2-day conferencing style workshops, and then we have 3 days hands-on practical workshops. As you can see from the images here, we have trainers, the young scientists are able to visit the labs and interact with these techniques and with these equipment, and at least be able to see and hear and learn from the experts themselves. Through this, we were able to register over 3,500 participants in 2023, with over 400 receiving hands-on practical training in molecular biology, genomics, bioinformatics, and LC. 40% were females, which is very, very exciting to see that we have a large crop of females who are doing this area. And the reason we are highlighting this is because in Africa, sciences tends to be a male-dominated area, but we see that we have gathered quite a good interest from the young ladies in this particular area. Our workshops were attended by students, by faculty members, by scientists, and even by industry players. And this shows that we're able to draw a wide base of representation. And of the 54 African states, we were able to draw representation from 49. So at least we had somebody from every part of the continent attending the workshops. We have also published our findings. And this is the paper that came out on the 2023 workshop. It came out last year. And last year's workshop is currently undergoing review by the editors. So we believe that our experiences, our lessons, our knowledge should be put out there for others to be able to read and learn and share the best practices. And that's why we are very, very keen on publishing our experience. We have gone beyond the workshops in the AfricaBP Open Institute, and we have established fellowships for genomic leaders for Africa. And last year we saw 4 fellows from the 4 areas of Africa attending this particular training. And there are various trainings that we have been able to develop with our partners. So if your institution is a partner, has signed an MOU with AfricaBP, then the students who are associated with that institutions will be eligible to apply for any of these fellowships, which are intense, hands-on. So some of them go for 2 weeks, others go for 1 month. And this particular one that is here was a 3-month training that happened between the USA and African institutions. So I'd like to leave you with some food for thought. One of the things that we as Africans have done very well is that we have moved into the genomics and bioinformatics space. However, we have to ask ourselves, how much recognition have we received as Africa for genomes and species that have been sequenced that are African nature? The truth is that very little has been done, and that is because the bulk of the sequencing is done outside the continent. We have to ask ourselves whether that is helping Africa or whether it is setting us behind. And I'd like to quote Thank God, who's the founder of Africa BioGenome Project and an AfricaBP co-chair, who says that an African-led project is not one where the PI has a primary African affiliation, but rather one where 100% of the research is performed on African soil. So Africans, we are included into publications, very good publications, international publications, but somewhere perhaps in the middle of a large number of other distinguished authors. And that is because the bulk of the work is not done on African soil. We have to ask ourselves, is it that we can't do it? Or is it that we don't want to do it? Or what exactly is the challenge? This is what we need to address. Because for as long as we continue allowing our genomes to be sequenced outside the continent, we don't have full access to that data. We don't have full recognition for that work. And at the end of the day, we may not have long-lasting benefits of that research being done outside the continent. So we are advocating that sequencing happens on the continent, and this is our contribution towards this effort, to make sure that we train early career scientists, we ensure that they get well-paying positions in African institutions. And we hope that these institutions will be able to invest in very, very good infrastructure. This will provide opportunities for promotions, for growth, and for development. So we also want to bridge any of the gaps in genomics and bioinformatics, and we can only do this if we work together as African scientists across the continent, because we have to determine curricula that will be practical and that will be long-lasting in our African institutions. So we want to remove barriers by impeding the growth of science in Africa by going fully open science. That means full access to training. You see that in this particular conference, we're not charging registration fees. We're not charging scientists for actually receiving in situ training in labs. So this is one of the things we also have to provide. Maybe the reason why we don't have so many bioinformaticians because of access to training, access to publications, where publication fees, they're expensive and they are proving to be a barrier, and also access to conferences, where conferences are charging registration fees, and these are barriers. So let's determine common curricula that can be developed to address this need. I thank you very much for your attention, and I invite you to visit our website and our social media handles on LinkedIn, on X, to be able to keep up with our developments in Africa BioGenome Project. Thank you very much, and I welcome any questions if there's time. Thank you.
Thank you, Professor Anne Mugai. So I don't know if anyone has any question for Professor Mugai, please. Any question, please feel free to wave your hand or put it in the chat box. Grace Ruth, you have a question? I see your hand is raised up. Okay, someone is asking a question. Makoba, well done. However, I have two questions. Is Togo country eligible? If yes, how are we going to benefit from this project? Professor Anne, do you want to respond to that?
All Africa states are eligible, and for them to participate in this conference, we need a point person in an institution that is running genomics or bioinformatics. So either the dean of the school where these courses are being taught, or a researcher who is sequencing or doing some research, they can get in touch with us through the email that is on our website, and that way we will be able to initiate conversations. And we are looking for key people across the different countries. So we welcome you to put us in touch with your professors, with your scientists, doctors, and we'll be able to get in touch.
Thank you, Professor Anne Mugai. And just to support what Professor Anne Mugai said, there is a practical session in Togo, University of Lomé, this week. And as you can see, University of Lomé is also one of the partners for this West Africa Regional Workshop. So I don't know, do we have Professor Kumi on the call to respond who manages things in Togo? Professor Kumi, are you on the call? Is Professor Kumi here? Yeah, I think it's muted. Okay, we're just going to go to the next speaker. Thank you, Professor Anne Mugai. We're going to go to the next speaker, the first session. We're going to be listening to Dr. Chika Kingsley. So let me see. Is Dr. Chika on the call yet? Okay, otherwise we'll move to the second speaker, who is Dr. Umar Ahmed. Dr. Umar Ahmed, are you on the call yet? Is Dr. Umar on the call yet? Okay, Dr. Kumi wants to talk.
Hey, Dr. Ebenezer, how are you?
I'm fine, thanks. Yourself?
Yeah, I'm fine. I didn't get I was following the I was following the presentation, but I didn't hear clearly the question. What was the question?
So the question is, I think someone asked if there is a site in Togo. So I mentioned that you are hosting a practical in Togo.
Yeah. We are having a practical session in Lome at the university level, but we are doing it at the National Institute of Hygiene. We call it INH. So the practical session is scheduled to be going on from Wednesday, Thursday, and Friday. 3 days. Everything is okay. I'm the one in charge of doing all the organization and all the program, all the planning. Everything is okay now. So I'm waiting for the one who has been shortlisted to participate to this workshop from Wednesday morning.
Thank you, Professor Kumi.
Yeah, so what we have to do, we will go from DNA extraction, RNA extraction, and the PCR, we will run some PCR also. So that is what we have planned to do for the 3 days.
Thank you, Professor Kumi. So I think this is useful to all the Togolese on the call today. All right, thank you.
Yeah.
So
Thank you.
The next, we have a couple of hands raised, quite a lot. Is Grace? Grace, can you unmute? Grace? Otherwise, we go to Stephen Ferrami. You have some questions?
Yeah, Stephen had asked a question online, and he wanted to know how institutions can collaborate. And I have mentioned that if they get in touch with AfricaBP through the email on the website, we will be able to get in touch and we can be able to collaborate. There's a question about eligibility for the workshop. All AfricaBP fellowships are open to institutions that are partnering with AfricaBP. So your institution has to have participated in the regional workshop or signed an MOU with AfricaBP, either one of these or both of them, for your institution to be eligible. So once your institution is eligible, the details of the criteria for the applicant are listed on the call. They're different for different people. Some of them are in genomics, others are in bioinformatics. And depending on that, people apply and we do the shortlisting. The shortlisting is done in partnership with the organizers, and people are then allowed to attend.
Thank you. Yeah, thank you, Professor Mugai. So we have a few hands raised up. Edom Moses, you want to ask your questions?
Yeah, okay. Yes, sir.
Go ahead, please.
Okay, thank you very much for this privilege to ask. We make mention that the Center for Practical, there is a center in Togo. So those of us that are based in Nigeria, so do we have a center?
Yeah, so there is, I think I'm going to allow Professor Mugai to respond.
Actually, thank God, this one is something that you can answer better than me. So
All
go
right.
ahead.
So yeah, there is each region of Africa has its regional nodes, which is in West Africa, University of Port Harcourt, and then national nodes. So any institution can kind of indicate to become a national node, and this is also part of the AfricaBP governance structure. And it's open, so anyone can indicate to be, for instance. So at the moment, for instance, people in Nigeria, in Ghana can also indicate to establish a national node, and they will be able to drive those processes at the national level. Does that answer your question, Edom? Yeah?
Yeah. All right. Yeah. Thank you.
Thank you. Okay. Mark Koba, I think you've answered your question already. Okay, so we're just going to move. Thank you, everyone, for the questions. We are going to move to the next presenter. Do we have Dr. Umar on the call yet? Umar Ahmed. Okay, Dr. Ahmed, so I'm going to make you a co-host so that you can present. Thank you for your patience because Professor Mugai came on board, and then we'll have to kind of try to follow the sequence of the agenda. So please feel free. Dr. Umar Ahmed is a senior scientist at the Africa Center for Disease Control. I've known him for a very long time, so it'll be interesting to hear what all the science has been doing, and this will be of interest to the African biogenome community. Dr. Ahmed, over to you, please.
Okay, thank you so much, Dr. Tangat. Can you hear me?
Yes, please carry on.
Okay, let me share my screen. Sorry. Can you confirm you are seeing my screen, please?
Yes, please. We can see your screen. Do you want to make it a slideshow, please?
Yeah, sure. Are you seeing my screen now?
Yeah, please go ahead.
Okay, thank you so much for having me this morning. My name is Umar Ahmed. I'm the Senior Technical Officer for Pathogen Genomics, coordinating genomic sequencing and bioinformatics across West Africa, and at the Center for Laboratory Diagnostics and Systems at Africa CDC. Today, I will be walking you through how genomics can be applied, to strengthen biodiversity surveillance and public health resilience in Africa. So biodiversity, you see, we have three different looping between biodiversity loss, emerging diseases, and climate change. These three components are actually what we call anthropogenic drivers that are caused actually by fossil fueling use, deforestation, and increased contact with wildlife, usually the cause of destabilizing the ecosystems that we have currently. And because of these interactive activities, these three looping that is happening, as you see in the cycle, a kind of cyclical looping between the three: biodiversity, climate change, and infectious diseases, is actually the cause of most of the emerging and re-emerging diseases that is happening. So this disturbing creates really conditions for pathogen spillover, and also novel diseases emerging and also threatening both diversity and human health. So I will start by telling you the One Health approach for the West African countries, since Nigeria is part of the West Africa. Generally, we have a lot of zoonotic diseases outbreaks in Africa, but particularly concentrated in the western part of Africa, which are actually the convergence or the cause of the humans, animals, and environmental health associations that causes all these pandemics and epidemics, diseases to be coming up. Particularly if you look at the West African regions, we do have kind of resurgence, emergence, infectious diseases like Lassa fever, Ebola, anthrax, Mpox. They are frequently been coming out by the day in the country, particularly in West Africa. So, and all this as a result of human-animal interactions, as well as the environment, that targets or that affected the agriculture, markets, and also the settlements where we are. So genomics can actually be used, to integrate these tools and also connect the surveillance across all the sectors, all the three sectors we mentioned. That is the One Health, humans, animals, and environment. So what is the roles of genomics in linking this biodiversity across all this species? So genomics actually can be used, for tracking pathogens, evolutions, emergence and re-emergence infectious diseases, and also identifying some reservoirs, vectors, and also transmission routes or patterns of a disease, and also spill over in the early onset or early warning signing from the environment when you carry out a kind of like metagenomic surveillance. That can also reveal how biodiversity alter pathogens dynamics, and also can support the One Health approach by integrating data, species, and ecosystems. So you can see the diagrams from the left side. You get to understand how these biodiversity landscapes, the looping systems that I showed previously in the previous slide, are connecting together, to cause the emerging and re-emergence of the diseases within the biodiversity ecosystems. So at Africa CDC, what we do, what is the genomic status at Africa CDC, and how does it impact, or what is the status across the African countries, actually lies based on the strategic plan that we have from 2023 to 2027. Africa CDC actually ambitions to develop a surveillance systems and an early warning signing systems through genomics applications to support the early detections, characterizing the pathogens, and as well as reporting the outbreaks as early as possible, and also apply clinical services on infectious disease and non-communicable diseases. The aim is actually to strengthen the clinical and public health laboratory systems and network within the continent. Particularly, Africa CDC developed what we call foundational genomics capacity within the African countries. If you see the first objectives, we started this in 2022 to 2023 to develop this capacity within the Africa CDC lab networks. And from there, we, encourage member states to have access to diagnostics, for effective detections, management, and control of pandemics. And also, we also do a lab mapping to identify the capacity of the labs, and also, what are their strengths in terms of surveillance, early detections, and also response to public health. For genomics, particularly, we actually developed the capacity of the member states, through molecular genomics and bioinformatics. We enhance their capacity by providing some genomic sequencers, reagent support, establishing linkages, sample referral systems, and all that. So when Africa CDC started, that was in 2017, there was nothing like NGS capacity in Africa. Based on the foundational genomics assessment that we carry out at Africa CDC, that was in 2018, 2019, when Africa CDC decided to establish the pathogen genomics unit. From there, we identified that in the entire Africa, we only had 7 countries or 7 member states that have functional genomics capacity, which means they can be able to sequence specific pathogens at a particular level, but they don't have, capacity to do the analysis. And what we identify, we are actually limited sequencing and data infrastructure, lack of skills workforce in the labs, and also the bioinformaticians are lacking in the continent, lack of policy and frameworks, no coordination of sequencing activities completely, challenges with respect to supply chain cost, and also regulatory challenges, when you send some reagent to some countries. These are the, the pre-assessment that was administered by the Africa CDC, that was in 2019, 2018, 2019, with support from some partners who were able to come up with, the problems. And those problems help us to establish through APRO network, we establish Africa CDC lab networks and regional sequencing hub. As you can see in my screen, we cluster the specialized genomics center. The western part, we have SGs, which are based in Nigeria, Redeemers University. We have Sanbi in South Africa, Eri in Kenya, and Chris in South Africa as well. So we cluster those countries with little or no capacity to some specific institutions that we call regional sequencing hub. For example, like in Morocco, we cluster Libya, Egypt, and the North Africa, to join Institute FASTA Morocco, because they have, sequencing capacity. In West Africa, for example, in Dakar, we, linked most of this laboratory in Gambia, Mali, Burkina Faso, Guinea. We link them with Senegal, to support them. If they have a little or no capacity, let's say they have pathogens in their country, and they are unable to do the sequences, they repair the sequence to, IPD for sequencing, and IPD could support them with analysis, even capacity building and support. All the rest of the countries, this is how we establish the networks and we leverage on the APRO laboratory networks and regional sequencing hubs. But we are particularly focusing on the national public health institutions and regional sequencing hub across the continent. And in 2021, the Africa PGI, the Pathogen Genomics Accelerate, really the sequencing of the SARS-CoV-2 genomic sequencing in Africa. And that really accelerates three major key pillars that we have on specialized centers and regional hubs, as you see. And also we democratize and expand the sequencing national reference labs, as well as the national public health institutions. We network across the continent with regional hubs and specialized centers. So what PGI basically does, the functions of PGI is to support the cross-border sample referral system. Initially, it started with SARS-CoV-2, and now we increase it to beyond SARS-CoV-2, including the use cases. We supported at that time 35 member states, with capacity to be able to monitor the variant of public health, the variant of the COVID for public health actions. At that time, we supported the shipment of 34,000, SARS-CoV-2 specimens in the continent. And we supported genomics and bioinformatics workforce. At that time, we conducted over 21 trainings. Over 600 trainees were trained across 52 African Union countries, and we focus more on the use cases that are frequently bedeviled in the continent, like the COVID-19, Ebola, monkeypox, and cholera. We also supported the countries with supply of equipment and reagents. We supported 32 countries with sequencers, 182 shipments of reagents to different countries, and also 200 kb of reagents, which 50% are actually procured, while 50% are donation from partners. And also we supported the quality assurance, the QMS, from the national public health institutions. So we had about 86% passing rates of the QMS for genomics quality assessment before the data can be uploaded to, sequence repositories. So these are basically the contributions of the Africa CDC and the African Union. We contributed 53 out of 55 member states with sequencers. And the sequencing equipment were actually 70% procured by the Africa CDC, 30% were donations. And we focus on 5 use cases: COVID, Ebola, Mpox, and cholera. And as well as we spend quite a number of money in, procuring some reagents across the continent. So what was really the gaps that was identified post-pandemic were limited molecular capacity, inadequate infrastructure and supply in most of the African national public health institutions, shortage of competent bioinformaticians and bioinformatics scientists who can analyze the data that was sequenced in those regional sequencing hubs. Wide varieties of pathogens, because there are dozens of public health events every day. You have, pandemics here, pandemics there within the country, so within the continent. And non-resilient and non-adaptive, unintegrated health systems that we have in the continent, and silo and fragmented surveillance effort were all the key challenges that we recently identified. So that was what brought to Africa PGI 2.0. Before, Africa CDC pathogen genomics initiatives, we are having 5 pillars that focus more on capacity buildings, sample referral systems, quality management systems, bioinformatics and genomics, training for all the public health and national public health institutions. But after identifying the gaps that we had, we sat down and come up with what we call the PGI 2.0, which specifically focus on 3 components: the surveillance, the laboratory components, and bioinformatics and data analytics, which are what we term integrated genomic surveillance for the public health actions. Without a very effective surveillance systems, there could not be effective laboratory systems that can carry out sequencing, and without sequencing, we cannot be able to have a quality genomics data that can inform public health decision. So this PGI 4.2.0 was actually focusing on 4 different priorities. The one is actually to expand on the foundational capacity. Since in 2018, 2019, we assessed the member states, we were able to understand their limitations, and we built, we built their capacity gradually, and then we were able to, start sequencing some specific pathogens. So we are now moving into expanding those, foundational capacity that we built across the member states. And secondly, we are actually implementing the integrated genomic surveillance systems for early warning sign and systems that involves making use of the wastewater surveillance system. We sequence even the wastewater to identify if there are, emerging pathogens or the pathogens that have the potentials for pandemic. We inform public policy on it. And then priority 3, we promote timely sharing of quality assured data through what we call pathogen data sharing platforms that is currently being developed by Africa CDC, where it is called AGARI Platform for Public Health Actions, where we ask member states to submit some data onto the platforms and, ensure we promote the timely sharing of the quality data for genomics. That will help country, to like a wake-up call for early warning signs and detections of pathogens with pandemics. And we also create and enable systems or sustainability mechanisms, frequently being carrying out advocacy, policy, governance, and also funding sustainability structure. So the 2.3, 2.0, the integrated genomic surveillance are actually based on response, samplings, analysis, and also the bioinformatics as well as the testing. So basically, if you look at here, is when we have the samples, we normally usually ask the member state if they have the capacity, they can sequence the samples, even perform some molecular diagnostic testing at the labs, and then from there, if they can prove they can test the pathogens, then it's fine. If they cannot, we refer them to regional sequencing hubs, regional sequencing centers to do the sequencing and also even perform the bioinformatics analysis for timely reporting. So these are basically the genomics surveillance workflow that we are carrying out at the continent using the PGI 2.0. So what are the role of genomics in One Health? One Health is a big, interconnectedness between humans, animals, and environment. And as I mentioned in the previous slide, that there are lots of zoonotic diseases outbreak in Africa, particularly in western parts of the countries. And these zoonotic diseases, the key ones are Mpox, Ebola, avian influenza. They are actually, genomics are actually being applied to those pathogens. For example, like Mpox, genomics help to trace multiple introductions that is sustained between human-to-human transmissions, and also identify spillover from wildlife and track viral evolution in Ebola case. Avian influenza genomic surveillance actually revealed the clade expansions and interspecies jumps, how they are interconnected, between humans, animals, and even the environment itself. Genomics can also have a reach, that can help in health, in tracking the health pathogens, and also strategizing through the molecular health insight. So it helps us in understanding the real-time genomics data, enabling early detections and confirmations of novel and emerging pathogens in the countries. So genomics for diversity and surveillance, if you see the previous slide, what I mentioned was actually a loop that is being cycling in what we call anthropogenics. And it's also the same scenario that we apply here, identifying the species as environmental scientists within the ecosystems, like the biodiversity ecosystems. We monitor, the spread of the pathogens and what are the shifts, and then also detect some environmental DNA from the wastewater, soil, air, and wildlife. This is how you genomics can be applied, in monitoring or carrying out some surveillance systems in the entire biodiversity ecosystems through non-invasive biomarker discoveries, as well as biodiversity databases, linking what is sequenced at this country, what has gotten in the other countries. That will inform where the disease are actually coming from, and we can definitely track using the applications of genomics in these ecosystems. So back to our discussions on how it was started, the PGI, and where we found African countries or African Union member states in terms of functional NGS capacity. There were seven countries that were able to have, functional genomic sequencing capacity, as you see in the map by the left. That is the pre-COVID. After COVID, in 2024, we were able to, expand the foundational genomic sequence that we found in 2019, to this level. Now we have 41 countries that have full functional genomics capacity in all the national public health institutions, or it affiliates like research institutions. And as well, currently we are trying to, support the remaining 9 member states that are not having the capacity at the moment through, partnerships with SLMs and many other partners to catalyze these capacity expansions in the national public health institutions, as well as the national reference laboratories. So this is what we have as post-pandemics as of 2024. And moving forward, these countries, we are able to actually sequence, at least we had 39 countries that can, be able to sequence the SARS-CoV-2. 16 can have the capacity to sequence the SARS-CoV-2. At least one other virus is either HIV, influenza, or polio, measles. And also we have 6 countries out of 39 have the bacterial sequencing capacity, like E. coli, cholera, and the rest. And we also have 5 countries out of the 39 that have malaria, sequencing capacity in the continent. So public health resilience will actually help, or through genomics, public health resilience can be strengthened through 3 different areas, like early warning systems. Genomics can support the early detections of novel and reimagined pathogens within the ecosystems of the biodiversity, inform targeted interventions as early as possible before even the pandemics can go, track AMR, identify resistant genes, in the environment, monitor trends, across pathogens, and that really help in informing decision-making based on the obtained data at early warning systems, tracking AMR, that guides the vaccines and treatments, as well as the policy responses to the countries. So in terms of the data sharings, what Africa CDC recently developed a continental framework in each of the subdivisions that we have at Africa CDC. And that continental framework actually encourages member states to promote timely sharing of data and also coordinated sharing of data. And that aligns with the African Union vision for sovereign, trusted data governance. In terms of ethics, equity, and engagement, Africa CDC ensures community involvement and culturally appropriate consent is sought before the data is shared to the public, encourages benefit sharing with data contributors, especially from low resource settings in the countries, and encourage fair, findable, accessible, fair principles of data sharings and open science practices across the member state to support basically transparency, reproducibility, and collaborations. So the key challenges thus far we found is basically infrastructure limitations. There are still limited sequencing labs, cold chain, supply, and also internet as well as the power supply are the key challenges in the countries. And workforce shortages, scarcity in trained bioinformaticians, even though we frequently train bioinformaticians, but they frequently left the countries, to other advanced countries. Lack of scientists and as well as data analysts. Sustainability issues, because of our heavy reliance on the donor funding and vertical projects. So, but however, we have some opportunities that could possibly help those challenges, which I itemize here. The use of mobile genomics platforms like portable sequencers, MinION, enabled field diagnostics and surveillance, applications of AI, enhancing pathogen detections, early outbreak predictions, as well as analysis, multi-sectoral partnerships like strengthening the coordination across health, agriculture, and environmental sector in a way of, one health approach that could really help. So what are the future directions for this? I thought of this for cycles, to encourage strong linkages with biodiversity programs like Africa BioGenomes, Genome, Africa VGI, linkages with One Health applications, human health environment, broadening One Health applications as well as doing what we call integrated genomic surveillance network. So the first is basically to foster strong collaborations and linkages between biodiversity programs. Second, establish cross-border and multi-disease sequencing platforms across the national public health institutions, and also promote the co-development of bioinformatics tools and data sharing, develop SOPs and that could be helpful within the countries, and expand genomics beyond outbreak response, so like climate-sensitive diseases, food safety and agricultural pathogens, wildlife health and ecosystems integrity. So the basic recommendations, the way forward after looking at the challenges, to strengthen the inter-country collaborations. As we understand, most countries are not really, collaborating with one another, even though they are neighbors. So facilitate sharing of sequencing infrastructure and expertise will help in strengthening collaborations in inter-country and regionally, promote cross-border data exchange, outbreak coordinations, and regional workforce, regional networks, as well as harmonize genomics protocols. For example, countries may decide to, develop standardized SOPs and metadata frameworks across sectors, align surveillance workflow between humans, animals, and also environmental health, and support the workforce trainings and infrastructure for data analysis. And one other thing is very important is, I think, retention. We keep training scientists across the continent for genomics, bioinformatics, as well as sequencing, but they keep leaving the countries for a better future, because there are so many other challenges that are there within the countries. I think the policymakers could, make use of their power, to find a way of retaining those bioinformaticians as well as the data scientists in the public health institution. That could really solve most of the problems and the challenges of data interpretations that we have in the country. So to conclude my talk, basically genomics is a cornerstone for One Health, particularly in Africa. So empowers early detection, surveillance, and response is the key. Links biodiversity monitoring to public health protections. So my call for actions is basically to foster cross-sectional, cross-sectoral and regional collaborations, inter-countries collaborations, support capacity building in genomics and bioinformatics, as well as align initiatives like Africa BioGenomes Project, GEOMBO, Africa CDC, PGI, national strategies, because most of the African countries nowadays are being encouraged to develop their national genomic surveillance strategies. We have about 10 countries now that have so far developed their national genomic surveillance strategies, and that really align with the One Health approach that Africa CDC are aiming at. With that, I thank you for the opportunity to speak on behalf of the Africa CDC and the Center for Laboratory Diagnostics and Systems. Thank you so much, and over to you if there's questions. Dr.
Thank you.
Tangot.
Thank you, Dr. Umar Ahmed. I know there are lots of questions for you, or we are running late, so we're going to allow maximum 2-3 questions. If you want to ask your question to Dr. Umar Ahmed, please do raise your hand. And also, if you have any question for him, also please do feel free to put it in the chat box, and he will respond via the chat box if we can't accommodate it during this meeting. Olushola Thomas, could you please ask your question, please? Or Yaya or Josephine, anyone can unmute?
Yeah. Hello, everyone.
Can you hear me?
Okay. Yaya, you want to go ahead?
Yeah. Good afternoon, everyone. My name is Yahya Said. I'm a master's student from Anhui Polytechnic University, China. So my question is that how can we benefit, we that we are not in the Africa, but we are from West Africa, but we are studying abroad, not within the region of Africa. How can we benefit from the African CDC?
Dr. Amar?
Thank you so much, Yahya, for these questions. Actually, Africa CDC, operates based on, the mandate of the African Union, which means if you are from the Africa, it means you have potentials to benefit from the training and capacity building by Africa CDC. But it focuses more of supporting the national public health institutions. So if you are affiliated with any of the national public health institutions in Nigeria, let's say like Nigeria CDC, ISJT, you will definitely benefit with the ongoing fellowships in bioinformatics and genomics, ongoing trainings in sample repairs, and many of the number of trainings that we often organize. So you're Africans, you have, the potentials, you can, of course, benefit from all of those trainings whenever it's advertised. Sometimes we do send those requests through the National Public Health Institution directors to request for nominees. And if you are lucky to be nominated, then of course you will be chance to benefit from the training by Africa CDC. I hope
Thank you, Doctor.
well.
Thank you, Dr. Umar. Dr. Umar, will have lots and lots of questions from you, starting from if you go to the chat box, starting from minute 10:30. Could you please go to the chat box and respond to the questions, please, and so that we can move ahead to the next point.
Sure.
All right. Thank you.
You're welcome. Thank you, Dr. Tan.
Yeah, thank you so much. Olusola and Malaki and Dr. Emmanuel, could you please put your question in the chat box, and Dr. Umar will stay behind to respond them so that we can be able to move to the next presentation due to time. All right, thank you. So I understand that the Vice Chancellor of the University of Port Harcourt is online now, or in the venue, or at the venue. Do we have the IT on site now? Control, are you ready? Or should we all carry on to the next agenda? So I think we're just going to carry on to the next agenda.
Dr. Tangut, no, we are ready, so you can just go ahead and.
Okay, is the Vice Chancellor ready? Is Control ready now on site? Hello? Victor, is control ready now?
Hello, can you hear me, Dr. Thank?
Yeah, I can hear you, please.
Okay, so yeah, the vice chancellor is available, so he
Okay,
can
we
go
can't
ahead and
see
put in.
we can't see the hall. So can you please stream the hall to the Zoom? So or you can
Yeah,
Hello, can you see, say you can't see the
we can't see the site, the control site.
well.
So maybe
Okay,
we move to the next. Okay.
No.
Mm-hmm.
Yes. Please, while we try to clarify that, I just want to say the Vice Chancellor of the University of Port Harcourt, Professor Owunari Abraham George Will, is here. He came in a couple of minutes ago and was happy to listen to the last presentation. So I just want to introduce him properly so that he can make his open his welcome address, because he's been quite busy for a while, so that you can take that and demo. Thank you very much. Prof, the Vice Chancellor, please, you have the mic. Yeah, all right, so please, I wish to call the Dean of Agriculture, Dean Faculty of Agriculture, to come and resume his seat with the Vice Chancellor. Yeah, we also have a Professor Valentine Tui from African Genome Center, Mohammed VI Polytechnic University, Morocco. Prof, please, can you join the visit? Yes, the Deputy Vice Chancellors have several official engagements along with the Vice Chancellor. The Vice Chancellor, because of his personal commitment to this project, had to break off to come here. So at different times, we expect that the Deputy Vice Chancellors will come in too, but we welcome the Vice Chancellor of the University of Port Harcourt, Professor Owunari Abraham George Will.
Thank you. Don't worry. I'll read here. Thank you. The Dean Faculty of Agriculture, our dear Professor Scholar from Morocco, the Dean of Student Affairs was also there, and then the Director, Professor Julian Suji, other distinguished guests were here present, my dear students of the University of Port Harcourt, and students from other institutions who are here present. Then those who are with us from online, especially the keynote speaker, who have just finished giving a very insightful lecture. Ladies and gentlemen, let me, on behalf of the Governing Council of this unique university, the management, the Senate, the staff and students, and our numerous stakeholders, happily welcome all of you again to the University of Port Harcourt, Cuba, Nigeria, for this annual scientific event at the instance of African BioGenome Projects. Distinguished colleagues, in February 2023, I was here to kickstart the maiden edition of the African BioGenome Project, Open Institute Regional Workshop, which brought together, just like today as we are witnessing, many of the brightest minds in the field of genomics. Assuredly, over the few next days, experts will delve into the latest advancements, innovations, and discoveries in the rapidly evolving field of genomics. Undoubtedly, genomics has revolutionized our understanding of life, its diversity, and disease. And it is an honor to be part of this community, driving progress with great prospects in improving human health. Distinguished colleagues, ladies and gentlemen, my welcome speech in AfricaBP 2023 workshop acknowledge that life scientists in recent times depend significantly on digital sequence data, on arrangement of molecules, on arrangement of nucleotide molecules in nucleic acids to resolve all manner of facts and evidences to have a better understanding of life and the basis of the existence of life. This development gave birth to the various levels of biogenome projects, which aim catalog and characterize the genomes of all sorts of African scientists, partners, with intention to achieve same goals for Africa as the Health BioGenome Project are strongly endorsed by university. Our greatest fulfillment is that African BioGenome Project stands a coordinated Pan-African effort to build capacity to achieve sustainable use of biodiversity for agriculture, for wealth creation, and ultimately good health across the continent. The University of Port Harcourt will always support AfricaBP to achieve its goal. It's indeed commendable that the African BioGenome Project, within 2 years of its establishment, published several papers in the Nature journals. Can we have a clap? To publish in Nature journal is not easy. In Nature journals, which is a great feat for that consortium. Updates from Professor Julian Osuji and alumnus and staff of the University of Port Harcourt is very encouraging. It's endearing that AfricaBP's Open Institute for Genomics and Bioinformatics have recorded such significant achievements in terms of capacity building, trainings, and fellowships, and recently commenced genome sequencing. University of Port Harcourt is pleased to be part of this phenomenon. We are open to partnerships with other institutions, bodies within AfricaBP Consortium. The university is glad to host this third regional hybrid workshop. We're indeed very happy to contribute our quota towards the growth and success of this worthy initiative. Distinguished colleagues, my dear students, and other guests present, we are here to learn. We are here to exchange ideas. We are here to interact with experts. We are here to interact amongst ourselves to gain knowledge and insight on this very important area of research. A research that will place mankind ahead in disease diagnosis, disease prevention, and disease treatment. In fact, for me, the future is now. I want to also commend all partners, sponsors, and speakers for their contributions for this year's workshop. To the delegates, those here and those virtual, thank you for being here. You will agree with me that without you, today's program will not be what it is. Your participation and engagement makes this event very special. I welcome you all to our unique Uniport. For those who are early researchers, for me, this is the way to go. Dedication, commitment, inquisitiveness, and you will have everything. This was the way we all started. several years ago, listening to experts, interacting with them. And by the grace of God, we are where we are today. So I urge you to keep this up and continue to engage with experts. Engage with experts. Once again, on behalf of the Governing Council, I welcome all of you, those on site and those who are virtual here, to the unique University of Port Harcourt for this very great event. As the Vice Chancellor of the University and the Chief Host, I hereby have the privilege to declare open this historical third West Africa BP serial workshop with the theme biodiversity, genomics, and One Health, biodiversity, genomics, and livelihood sustainability in West Africa. Let the discussions begin. Thank you.
Thank you very much, great Vice Chancellor. That was a great speech, as expected. Please, while we consider that speech, please let me recognize the presence of Professor Brilliant, Director of the University. Professor Brilliant, please, you can also step up. I want to also recognize the presence of Professor Chima, the Dean of Current Affairs. So this is a time when the university is quite busy trying to put things together. Now the students are on holidays, and the people, important personalities in the university will be dropping in gradually as we move on. Now we will need to take just one or two comments because Vice Chancellor will need to go back to his office and attend to other things. We will need to go and take group photograph with him when he's going to leave, or we'll leave that to his, let it be at his own discretion. If I want to listen to one or two more, because our Vice Chancellor is the medical field, is a professor in the medical field, and this program is loaded with medical genomics, medical bioinformatics, and all of that. So we'll take a few comments while we'll bring the DC to decide what next. All right, so yeah. Okay. My dear students, you know, this is the reason why we are here. We are here to learn. You know, so when I come to conferences, workshops, and I see you, I feel happy because it means you know why you are here. The university is a center for knowledge, and we are here for knowledge. So do not ever think you are wasting time. You're not wasting time. Whenever they call you to events like this, it is for you to interact with experts. I'm repeating it because I'm seeing some young people here who are early researchers. Please keep it up. It's very important. I'm proud when I see you. And I know you are here not just because you want to hear, but you want to learn. You want to go with something. And believe you me, this is an area of research that is not easy. It's difficult. But with the experts around you, they can simplify things for you. This is the future of research. And I'm very glad that I'm seeing some early researchers, my students who are here. I want to thank you and to say that please keep doing this. Keep coming, keep learning. And then your tomorrow will be better assured. Thank you and God bless us all. I think we can move on. Thank you, Professor. Dr. Tango,
yes, Professor Masuji.
All the
chairs, Senate committee members, co-chairs, and officials from the Africa BioGenome.
Outside, I mean, most like.
Yes, yes. Please, we'll go outside to take a group photograph, and we'll take 10 minutes to sort all of that out. When we come back in, we'll now continue with the program. Thank you very much.
Thank you very much, Professor Soji. To those online, so please do give us 10 minutes so that those on site can go and take the group photograph as per the program of events, and then we'll return to the presentation of Dr. Chika Owama from NIMA. I'm gonna is Dr. Owama on the call now? So I'm gonna spotlight him.
Yes, I'm here.
Okay, just a moment, please. So while we wait for, so you can just maybe put your screen, you can start sharing, and then or put on your video so that we can spotlight you and wait for them. So hopefully in the next 9 minutes, 10 minutes, they'll be back. Do you want to share your screen, your presentation, the first slide? Just put it up so we can. I've made you a co-host.
This is SNTF. See, don't drop it on this thing because you don't.
they've concluded with the group photo. Thank you everyone for your patience. So we're gonna go to our next speaker. So again, I'm gonna post the agenda in the chat box so that we all follow the agenda. We're running late, and thank you for your patience, and we'll try to meet up with time. So our next speaker is Dr. Chika Owoma. He is the Deputy Director and Head of the Microbial Department at the Nigerian Institute of Medical Research. Dr. Owoma will talk to us about multi-omics approaches to understanding host environment interactions in West Africa. Over to you, Dr. Owoma.
Please
Are you on mute?
Can you hear me?
Oh, yeah, we can hear you. Please go ahead.
All right. Good morning, everyone. And depending on where you are, it could be a good day to you. Glad to be here and to discuss a topic dear to my heart. Thank you for all you do, Tango and the team. I'll be speaking on multi-omics approaches to understanding host pathogen and environment interactions. I hope to just bring a few things for us to discuss. I'm looking forward to it being something that will stir us to think of what we need to do for ourselves and for our country. So, Nigeria is this amazing giant of Africa, a bit of contradiction in different ways. There are several estimates of our population, no real figure. But the other thing that's interesting is that births keep coming. If we go by this 5.2, we will keep increasing. And that means we need to really do some work to make sure we secure ourselves, our future generation. I tried to put up the top causes of death in Nigeria, both for males and female. You still see that infectious diseases are still top up there. But we also see non-infectious disease, stroke, coming up. Ischemic disease. And these are things we also need to talk about and handle. Now, I'll quickly do a bit of introduction to multi-omics. I picked some of these from open websites, so people can also go and pick this mostly from open websites. So the first thing is, some people call it multi-omics, some people have a dash, some people just call it integrative or pan-omics. The important thing really is that it's about a time when we are pulling data from different sources. It can be genomics, it can be epigenomics, transcriptomics, microbiome. We are pulling them all together in such a way that we're able to analyze data and be able to find out what is going on. We're able to find out novel associations when we are looking at different things. We're able to pinpoint and, more importantly, elaborate on relevant biomarkers. Particularly when we look at the genotype, the phenotype, the environment, we know there's this statement we often have: nurture and nature determines who you are. So that's where system biology comes in. It's kind of the thing that helps and gives birth to multi-omics. And we need to begin to look at it in a comprehensive way and know that everyone is important to this project. So in trying to just clarify a bit, the genomics may be the DNA. You look at epigenomics, some changes that are not linked up to changes in the DNA. And then the microbiome, we find that everybody, we're a compendium of different things living in us, gut microbiome, different things. And then the transcriptome, the metabolome. So there are different areas looking at the proteins and all of that. And all of that determines the person and determines your status as someone who is healthy or diseased. And I like to call this a snapshot of the organism. So it's as if you take the organism, cut it into half and have a snapshot across the organism within its environment. So if you're a biomedical scientist, you may be looking at the gene first, but the clinician may be seeing the phenotype first. So depending on wherever you are coming from, we all need to be able to look at it, position the organism in its environment, and then we begin to see what is the DNA saying, what is the epigenome saying, the transcriptomics, the proteomics, and then take into cognizance that we have several organisms living in us, whether it's the gut microbiota, whether it is vaginal microbiota, whatever it is. We acknowledge those things, and some of them are actually important for health of the organism. So, and when you look at these host pathogen environment considerations, there are several things to look at. But one key fact is that the interaction between these three determines health or disease. All three. In fact, that's why at times some people will tell you that the first kitchen, the first hospital is your kitchen. What you cook, how you cook it, how you eat. But you know, when we also, I have a lot of students and people coming to meet me, oh, I'm trying to work on this, I'm trying to work on this. And then you say, oh, someone that has HIV, someone that want to look at the DNA and know what is happening. They forget that there are two sets of people, organisms involved, the host and the pathogen. So if you're talking about the genomics, is it the genomics of the host or the pathogen? If you're talking about the proteomics of the host or the pathogen, all of them are involved. And that is where multi-omics is very, very important. So you are looking at the DNA, the mRNA, the proteins. Is it of the host or the pathogen of interest? And then there are other complications. I don't want to take this too far. I know not everyone is a genomics person, but there are people we call mosaics. I use a very simple example. Most of us know the sex chromosomes, XX for women, XY for men. Now that XX, the woman gets one from the father, gets the other one from the mother. But the cell, in trying to keep things normal, just activates only one. So in your palm here, or in your skin here, you may have the X from your father activated, while on this other arm, it may be from your mother. So you are actually a mosaic, and different things can happen. And that's where people start looking at, oh, in the same organism, look at a cancerous cell versus a normal cell, that one that doesn't have cancer, and see what are the differences you can pick. And that's why some people may have a particular cancer in a particular cell, not in another place, because those cells may actually be different from each other, even though being in the same organism. When we talk about the environment, it's also equally diverse. Behavioral practices, lifestyle, what are your exposures? So we may be in Lagos and we have a lot of bustle. People don't sleep so much. They wake up very early and they go back home very late and they wake up very early again. And over time, it begins to drain them. But in pH there, you also exposed to a lot of things. I know about gas flaring, and a lot of that is in the air, and you have been breathing it for so many years that you have been living there. How does that impact you? And then there is the other complexity for us in Africa, where we have co-infections. This is just a snapshot from a kit I developed for febrile illness, where people, you will think, oh, people that have a fever is malaria. Most of the time, it's not malaria. And then you see in sample 14 and 16, people that were supposedly with streptococcus, and they had streptococcus and dengue, and streptococcus and salmonella. So imagine treating those three people, 14, 15, and 16, for streptococcus, and 15 is getting well. The other two are not getting well, because you don't know that they had other co-infections. And in this case, you'll be talking about the genes or the genomes or whatever of two different pathogens, the host, and so a lot of complexity will come into place. Now, some of our cross-cutting tools don't apply. So there are many nucleic-based methods. These are just the same pictures, but in different ways that can apply, that I can use for genomics, epigenomics, or transcriptomics. You can use mostly nucleic acid-based methods. But when you talk about the proteins, other things come into place. So you may have to use GCMS, mass spectrophotometry, and a lot of other things to look at the proteins. And those people you will need may be different. The biochemist and different people will need to come in. So we are pulling data from different sources, different areas, each one heavy in itself. Genomics data, if this whole genomics, can be quite heavy. But think about the spatial geometrics of a protein or something. It can be something else on its own. So all of this information, when you pull them together, you need to make sense of it. And while you are trying to make sense of it, are you making sense of that from the organelle? Are you making sense of all this data from just one cell? Are you making sense of all this data from just an organ in the organism, or from the entire organism? And that's why people like Dr. Man has gone ahead of me that spoke earlier. Bioinformatics, they are very important to try and analyze these things and see, oh, where is the environment coming in? How are these things putting together? What is the combination of these things saying to us about how we're determining disease or determining healthy status? So there are several other challenges that may not be linked to your data and your source of information. As simple as your sample, how we are picking the sample. We recently did a study we called Huglosin, and it was something about after we saw data from COVID-19. In fact, I said with my DG telling me, oh, someone needs to look at this, my PA. My DG then said, I travel everywhere with my PA. I've come down with COVID twice. He has never once been had come down with COVID. And we all laughed about it. But I went back, I went to check. And I saw that there were genes that were influencing COVID-19 presentations. And we began to look at it. So we tried to pick samples from the northern Nigeria, Port Harcourt, for all the minorities, southeast, southwest. In picking those samples, you could actually say, oh, I've picked the North, I've picked Hausa, Fulani, I've picked Igbo, I've picked Yoruba. But how diverse are they? So before we recruited you, to be sure you were representative of that population we are recruiting you for, we had a caveat. Both your father and your mother must be from the same tribe before we took you in as someone representing Hausa, Fulani, or that. So those kind of things can actually influence things. There can be different kind of presentations. People that have asymptomatic or symptomatic infections. How does all of that in your data influence or bias you? So those are other challenges that you may have to look at. Ultimately, we want to go from a time where we are thinking of one treatment is for all to a place where we have personalized treatments. And that's where every hands have to be on deck to bring in the genomics data, the proteomics data, bring this all together so that we can actually treat people very well and be sure that, we say treat them right the first time they get well. So I want to bring up some examples before I zero in. I'll throw up a few case examples for us to think through. Yours may be different based on what you are doing. I saw some of the charts. Some people are also in plants. Yours may be different, but just to make us think a lot. And then I try to zero in on one before I wrap up. So this is a screenshot from one of our assembly tools for HIV. We sequence HIV for treatment of the people who are living with HIV, and they can use them to change their drugs. Now, I don't know if you can see the mouse. You see here, we have the standard HIV. We have this person we just sequenced that we are assembling the data has inserted a full codon. This codon is not in the standard HXB2, but is in this sample. We have seen people that have inserted 2 codons or deleted 1 or 2. What does this mean for the person? It could mean, oh, there's a protein that is not available. If that protein is what maybe a drug is looking out for, or an epitope, or a target that biomarker is going to work, or the drug is going to work through, the drug may fail. What does that mean for people? And then that means this can also be there in the system. You may not detect it. If, for instance, your detection kit is based on this area, it may miss this. And then you may know, you think it's going down, or you don't know that it's expanding. You may not know what is going on. And then we had a colleague we worked with in the University of Lagos. She was working on endometriosis. She picked 4 SNPs that were supposedly associated with endometriosis, I think among Caucasians. By the time we finished running it among Africans in Nigeria, Nigerians in particular, only 1 was relevant for our population. For 2 of those SNPs, both case and control actually had the same genotype. And this brings up to 4 that where we in Africa, we can actually take advantage of the diversity of African genome and be able to fine tune. When you go to all this ClinVar, they will say, oh, this mutation is pathogenic or not. We can actually use our genome to actually fine tune and clarify, the impact of these mutations. I've had people that approached me for us to do some genomic studies. They were thinking that people that had successful surgery, some of them didn't wake up early, not because the surgery was not successful, but they suspected that they were reacting to the anesthetics they were given. Another ENT doctor reached out to me, can we do something on ENT? That they had a 50-something-year-old woman who was always having ear pulse, things running from the ear. And the same thing is happening to her children and her grandchildren. And these are examples of different work, what we need to do for our nation that nobody else will come to do for us. Now, I'll try to put up the Hugo Sin study. It was during the COVID-19, and I saw this paper in Nature from the COVID-19 Whole Genomics Initiative. They listed out about 13 loci in the human genes that influenced COVID-19. The influence was different. It was, they put it in three. It was whether you're going to be infected, whether you're going to be critically ill, needing ventilation, or whether you're going to be ill and hospitalized, but not that critically ill. And some of them cut across all three, some influenced just one. By the time we finished gathering these samples from Hausa, Yoruba, Igbo, the person that worked with us from Port Harcourt did a very good job. We had almost all the different tribes, Isoko, Ijaw, different people coming in. And by the time we looked at this, we saw something overwhelming. Most of the alleles that brought about the effect, whether it was to make you ill or be infected, were not in our population. We actually were saved by our genome. And we laughed about it and said maybe something like this have happened in the future and clear those people susceptible. The most important of all of this was the last one there. We did not have a single person having even a heterozygote allele for the effect. By the time we worked with Lagos Biobank to pick samples from those who were Asians, who were in the what's it called at that time, who were isolated and treated, we began to see those who had 1 copy, 2 copies of this same, the last 97700 RSP, meaning something that we didn't have this, and that was why we were mostly running about, going about, COVID infected and still asymptomatic. I remember I was going to travel one December for a meeting, and I just got to I was about going to the airport, and I just oh, I've not seen my COVID-19 report. I sent they call it the lab. Oh, give me my report. What's going on? It says, sir, you are positive. I said, hello, excuse me, I'm about traveling. I packed my bag. What are you saying? Unfortunately for me, I developed one of the kits for COVID-19 at the time. And they told me, sir, it was your kit we used. It says you are positive. And I had to go back home and said, okay, it's all right, I'm not traveling. So some of those things, we have to begin to look at them. Now, you must not be a very big person to look at it. I tried to put this slide to show you that even from your MSc, PhD, you can. I had 2 of my PhD students who are working on infertility, but they are tracking, cutting across like a snapshot of that individual I mentioned earlier. They are looking at sperm counts. They are talking about infertility. They are explaining infertility where every other thing is working. By the sperm count and all that, you should actually be able to have your babies, but you're not having babies. They are looking at anti-sperm antibodies. They are looking at key infections. They are looking at mutations at different time points in the infertility pathway, whether it's for the man or the woman, and then trying to come up with something. It's an example of what you can do, even in the sense of multi-omics, to do something. So this person just finished defense for her PhD. She looked at serotonin and effect on depression. And then she looked at the long and short. She did CMV analysis. She did some SNPs analysis. She measured the hormones themselves. She measured some testosterone and different things that were also linked with them, and tried to come up with which ones are influencing depression in our people. Now I try to focus on HPV as I end. So HPV is one of the infections associated with the cervical cancer. Human papilloma virus is about 9.7% in the last most updated document I saw online. And I think I like to mention this. Kebbi State is the only state I saw that already had a good report of their cancer program treating their people. We published this in Frontiers in Oncology. It was during the COVID-19, the sample collection kit we sent to the north didn't come back. So we were able to just report on the southern part of Nigeria, trying to show that we have much more of other high-risk groups. So even though 16 and 18 are known to be much more associated and cause about 70% of the cervical cancers, we have a lot of people infected with other high-risk groups. If you look at this, we are seeing 16 and 18 are not the most prevalent. But when you look at, so the example we use here, one of the sites we used to recruit people was actually a site doing infection, what's it called, clear inspection of the cervical, of the cervix, and to know whether you have had changes. So we're able to link it up with, oh, were there changes in the cervix for you, for those who could give us their results? We saw that now you see 16 and 18 are coming up higher with those who had VIA positive results. But there were others who had other strains, who had other strains and had VIA positives. So we need to look at this. And I'm going to take this further when I look at the Cancer Atlas project. The other part I wanted to mention here also is that in terms of bringing in the environment and the risk factors, if you take alcohol, your risk was 1.7% of being infected. If you smoke, it was 2.6. By the time you smoke and drink, it was about 2.4. That will be the odds of you having HPV infection. So the environment, the lifestyle also counts. Now, I'm going to put this up so that we can also try and look at this later. Two key papers that use multi-omics to investigate HPV and cervical cancer. I particularly like this. From their graphical abstract, it's very exciting. So they had this number of patients, 106. They were able to capture the virus, looked at the RNA. You could think of transcriptomics. Looked at, oh, where is the HPV integrating in the host cell? They found that the integration point was non-random. It wasn't random. So the HPV were choosing places that were always replicating to integrate itself. And then they also looked at which of them was productive of oncogenes of the E7, E6, E7. They were able to find out that, oh, those that were productive were the ones that were going higher. So you can imagine you may have two people who have CNS1 or CNS2. One person proceeds to cancer, another person does not go on to cancer. It could just be their host DNA. Maybe one person has good DNA repair gene, another person doesn't have. So these are different things. It just gives you a good example of how we can use multi-omics and tease out issues and begin to even discover new drug targets and new areas we can actually zero in and be able to make a difference for our people. In wrapping up, I wanted to just talk about some of these challenges that go against some of these things. I'll try not to be very, but you know, we have health personnel interrelationships where people are fighting about who is the head of the lab or who should be in the lab. When we talk about this genomics, how many clinicians understand it? How many clinicians will use the genomics result to treat their patients? Is government supporting genomics research? You know, the government, they can easily make a road, 10-kilometer road. People will see, oh, this government is working. Rather than invest that money in a genomics project that nobody will see anything and people will think they are not working. Many institutions may want to buy equipment, but not the research. Some of those equipments, they may not even have the personnel to use them, and they may be in cartons years after they have been bought. How do we build capacity to utilize this data and network? How do we work with colleagues to train? You know, the truth of the matter is someone has to generate the demand. The clinician, the pharmacist, they have to generate the demand. Government has to support these things, develop things. Government is not being used by the government of the country. Who else will use it? So we need to look at all of this. More importantly, use a One Health approach to look at these issues. Just in making a few recommendations, we need to collaborate. We need to identify low-hanging fruits. Look at how we can build capacity for omics. More importantly, not just to generate the data, to interpret it And that's where I will emphasize what Dr. Omar has said before. And that's where it's important, because you see, you can generate this data. Anywhere can generate data now. But it's your ability to interpret it and be able to use that to produce, to change practices and generate products. And that is what is like more than $1 billion industry globally, which we need to key in as Africans. I want to thank you all. And I noticed something Omar said. Many of these people here were my interns or students at some point. Over 90% of them are out of the country. And we hope that some of them will come back. Someone is about wrapping up their PhD, and we're trying to see how they can come back and also work with us. But I'd like to share this saying with you. It keeps me going, even in spite of all the challenges, that a dream is not just what you see while sleeping. It's something that does not let you sleep. You remember all of this work we need to do, and you are sleeping, just wake up. Oh my God, we're not done this. We try to do what we can. And if every one of us from our own aspect put in our bits, Nigeria will be for the better and Africa will be for the better. Thank you, everyone. And thank you for inviting me. Over.
Thank you, Dr. Chika. What an insightful presentation. I felt like we are all fired up. That moment when you are fired up and you feel like the whole activity just got started. So in the interest of time, Dr. Chika Owama, we're going to just take 2 questions. Then you have the opportunity to respond to other questions via the chat box. I'm going to take 1 question from the audience on site and maybe 1 from those joining remotely. So we'll just start from one of the audience joining remotely. Do we have any question? We have someone raising their hand. Then do we have any question? Actually, so these are just comment, people reinforcing what you mentioned. So Josephine Don Bosco, could you please ask your question and unmute?
Okay. Good. Could you hear me?
Please carry on. We can hear you.
Okay. Good morning, everyone.
Good morning.
Good morning.
My name is Josephine from Benin. I'm a master's degree student in genetics, biotechnology, and natural resources. And my question goes directly to Dr. I would like to know, you were talking about being in a kind of mosaic, talking about our genomes. I would like to know if it's only about men or is only about women or is about men and women. And apart from that, when you were talking about having one of the chromosomes like S activated, how can we know that the chromosome X that is activated comes from the father or maybe the mother? And do we base ourselves on that in order to treat the patient, or we just try to carry out the 2 genomes of the person? I'm talking about women. We just carry out the 2 genomes and base ourselves on that in order to treat them. Thank you.
Okay. Do we take both questions before I answer?
Okay, you just answer this one and then we'll move to the participants on site, please.
Okay, thank you so much for the question, Josephine. I just try to use that as an example I think most people will relate with, because I know most people know about X, Y, XX, So it's not really important trying to find out which of the X is causing the problem and which cell has it. Most of the time, what they do is, if a person has a cancer, they may want to look at the cancer gene, maybe pick a tissue from the cancerous tissue and or organ, and then compare it to another cell from the same person, trying to find out what went wrong or what it's there. I spent some time at the Emory CDC, Emory University. They were even sequencing dead fetuses, people that had abortions. They sequenced them, did microarrays on them, Affymetrix, big arrays, and tried to pull together the data and find out where the mutations were. So when the patient, the parents come back again, they know how to treat them. So it's not really necessary to find out which one is causing the issue or which one, but just that to understand what is going on, that we actually mosaics. The cells, you may find out why is someone having, what's it called, cancer of the kidney, or some not cancer of the stomach. Why is someone having this other cancer, not this other cancer? So some of those things are where it comes in. So in the terms of the multi-omics, you look at what you can compare and be able to help us get some information that is generalizable that we can use to also influence policy, practices, and products. So that's the sense we will use that. I think that's, yeah.
Thank you, Dr. Chika Onwuama. There are some questions on the chat box, so please feel free to respond to them. Do we have any one on site who would like to ask a question, please? Just one question, so that we can move to the next speaker. Control, do we have any? Okay, go ahead, please.
Yes, my name is Dr. I want to first of all thank the presenter for such a wonderful and very expository lecture he's just giving. My question is in terms of government policies and awareness. I don't know, I know that this is the second time that we're having this meet. I don't know, are they biobanks in Nigeria? That's one. Then two, how do we drive this message to policymakers, to people like you rightly say that politicians will be more interested in road construction than in doing the development? How do we get at least to start to look like as if we are on track to getting some things and domiciling in this your lecture, you know, we would have been looking at things one-sided. When you talk about the genome sequence, you're looking at the HIV, this you're looking at only one side, the pathogen, you're not looking at the also. You understand? So there's that gap between the host and the pathogen itself. How do we push the drive, the government policy drive, in terms of this research and development? And again, like I said before, can't we develop our own biobanks here in Nigeria? Thank you.
Thank you, thank you. I see it just confirms you engaged well with. At some point, I was wondering if you were hearing me. So biobanks, there are biobanks in Nigeria. In fact, in Naima here, there's a very big biobank. It's 3-story. 3 stories, and it's large. It's just been established. We're trying to work with different people to set it up. I know IHB and also the biobank that worked with the H3 Africa, and then I know on another project, Dr. Ndodo will be talking about the environment for genomics. I know we're also working on the genomic consortium to set up a biobank, but we can also start something. For instance, if you're a clinician or an expert in diabetes, you can work with the people and start something. Because there are, you know, we can't get to that extent in one day. Thinking about the UK Biobank, they'll do a study. They had 9,000, I mean 10,000 twins. Where do we get that? They've been compiling data of twins. And then someone just goes to the UK Biobank, gets the data, and be able to do the sequencing and do whatever they need to work on. You know, but we can start small. That's one of the things I'm an advocate of, you know, being that we are still doing advocacy. Each and every one of us have someone in government that we have access to. Keep talking to them. But let me surprise you. It also starts with us all. I don't know if any of us have had someone, a relative that died, or a relative that has money. I come from Anambra State. And someone recently told me that someone was burying someone and killed 1,000 cows. Let's say, for instance, that person died from diabetes. Instead of that 1,000 cows, you could have done an endowment to research on that diabetes. If we begin to engage with ourselves, our people, someone, that's how they say this abroad, some of them. Someone died, oh, they have money from this. Okay, let's set up something. Let's begin to research on this, that no one else will die, or I will help someone else that has this. So instead of going to all this, I'm in Lagos now, Yoruba, you know, you can go to a party, you can receive different gifts. Oh, these people will give you, these people will give you, instead of all those different gifts, some of them very expensive items. You can pull that money together, set up a research, a philanthropy. During COVID-19, the kit I developed for SARS-CoV-2, it was sponsored by a philanthropist, the co-founder of GTB. 20 million was given to us to develop that kit. So we can all start this and start small and keep advocating. And in ending this, I'll say we are all members of a team. I mentioned when I was talking, there's a place you need the bioinformatics. There's a place you need the biochemist. There's a place you need the genomics. I just finished over this on Saturday, finished sequencing, we did 16S sequencing for trying to look at microbiome from oral, people that were living in HIV, swab their mouth, that people that had sores, try to look at what other microbiota is in their body that is making those people have sores, different from others who don't have sores. And we took their, extracted the whole DNA, RNA, and we did 16S sequencing to find out which other things are there. I did the genomics work. I finished the sequencing work. I sent it to the microbiologist. I said, I don't know what people are speaking my broken here. I could not understand the different genomes, different things. I just said, I finished my own sequencing, see your result. I sent it to them, and they're looking at it. You know, so all the teams we come together. It's only in Africa we begin to think we are, if you show your proposal to the next person, they will take it away. No, synergize, even abroad, even if you are the person's enemy, they will work with you, because working together in a disciplinary team, you will get the big multi-million-dollar grants. Thank you, everyone, and have a wonderful meeting.
Yeah. Thank you, Dr. Chika Owama. I have so many things to talk about, and also but to kind of support your comment. But in the interest of time, I really have one responsibility now, is to get us back on track on time. So I'm just going to move to the next presenter. Thank you, Dr. Chika, for coming. I really appreciate, and I hope we can continue this conversation. Gloria, I know you are raising up your hand. Would have to kind of go to the next speaker. Could you please put your question in the chat box? Then Dr. Chika Owama would then pick that up via chat. Thank you. The next presenter, it's supposed to be Dr. Nnemeka Ndodo, but he sent a message this morning saying that he would want to present tomorrow due to an activity he needs to attend to. So we're going to move to the next other presenter, which is Dr. Ashraf El Alali from Mohammed VI Polytechnic University in Morocco. He's going to talk to us about the African-based databases to improve agriculture, One Health, and biodiversity. Dr. Alali, I'm going to make you a co-host. Is Ashraf on the call yet? He's not? Oh, he's not on the call yet. So we're going to move to the next. I can't find it on the call. So we're going to move to the next presenter, which is hopefully he'll be able to be back. So do we have Sarah Gaidi on the call? Sarah Gaidi is an oral presenter. Sarah Gaidi, yeah, I can see Sarah. Sarah, could you please unmute and then share your screen and make your oral presentation, please? Sarah Gaidi, you can now unmute. Sarah? Can you unmute, please, Sarah, if you're talking? Okay, I think we'll probably have to go to the next presenter. So do we have Mamodu on the call yet? Yadi? Yeah, Mamodu Sambo Yadi. Could you please unmute? You are now a co-host. You can unmute yourself. You want to make a presentation of your oral talk? Mamadou Samb Yadi? can see Mamadou, but he's not. Is he on? If you are talking, you're mute, Mamadou. How about Okechukwu CL? Okechukwu CL. You're not a co-host. You can.
I am.
Okay. All right.
Good
Please
afternoon,
carry on.
sir.
All right. Good afternoon. Please carry on and share your screen and make your presentation.
Good afternoon.
Can you please share your screen?
Yes, I'm on it, please. Okay, one second. So I'm trying to share my work.
So while Okechukwu is preparing to share her screen, can Ezekiel Olowalaju start preparing because he'll be presenting next? Ezekiel Olowalaju, please start preparing because you'll present next. Okay, Okechukwu, are you ready? So now we lost her again. Is Ezekiel ready? Ezekiel, you're now a co-host. If you're ready, could you please present? Can you hear me? You're on mute if you are speaking. So I think, okay, this is the one break time. Okay, Chuku, are you free to present now?
Okay, I can share my slide.
Do you want to send me your slides so I can share it on your behalf? Is that okay?
Yes.
Okay, can you send it via, do you have it on a Google Doc or just, so send it to this email address, please. Can you see the email address? I just sent it to you in a personal Zoom chat. So do you have it on a, yeah, just send it as an attachment, or if you have the Google slide URL, you can send the URL, please.
Okay, let me share it there.
Have you sent it yet? Okay. Chikodi, have you sent it? Okay, Chuku. So I think we can now, at this time, see if we can try to maybe have an early launch. That way we can be able to kind of keep up with the time on the program of the agenda. And hopefully, Chikodi, are you able to still present? I can't see your email yet.
I have sent it. I've sent it.
You've sent it. All right.
Yes. Yes, have you seen it?
Yeah, I've not seen it.
Did you see it?
No. Yeah, so I think we can just have an early break and then return back in about, so I think as per the program, the lunch is 1 to 12, 1 to 2 rather. So we'll have an early break and then return back at in the next 16 minutes, if that's okay. That way we are then 30 minutes early. So that would be at 1:30. Is that okay? And then we can that would align us with the program of event and ensure that we keep up to the schedule. So maybe Chikodi, can we pick up your presentation as the first presenter, and then is that okay?
Yes, it's okay, sir. Thank you.
Yeah. So let's go on.
I said it's okay, sir.
Yeah, let's break out for the break for the lunch session, and then we'll be back at 1:30. That will be in exactly 16 minutes. And hopefully we'll be early for the other session, and then ensure that we keep track or keep kind of aligned with the whole program for the rest of the day. See you in the next 16 minutes, please. That will be 1:30 GMT +1 or 1:30 Nigerian time or West African time.
Look, you're not actually going to get anything done by watching another YouTube video. In fact, you're literally only procrastinating more. What you need is something I like to call a GYST week. That stands for Get Your Together Week. Because let's face it, you need to. All right, so what does it actually look like to dedicate an entire week to just tackling everything that we've procrastinated in one fell swoop? That means finances, folding laundry, calling your mom back, personal projects, you name it. Life would be so much easier if we could just get everything done and out of the way for good. Does all that sound overwhelming? It's because it probably is. But here's the secret. It doesn't have to be. Just schedule all of your tasks into a GYST week. All I do is take like 8 minutes at the beginning of my week to brain dump my entire to-do list, and this tool automatically sorts it all out for me. It'll schedule my tasks, set reminders, and the best part is, if I miss a task, it'll automatically shift my to-do list so that I don't have to keep staring at what I missed. Basically, it's the only tool I've found that combines the best parts of a calendar and a to-do list and wraps them all into one using AI. If you're tired of feeling like life is a never-ending game of catch-up and you're tired of being stressed out over all of the little things that life throws at you, you need a GYST week. Click the button below for the AI personal assistant that'll make this whole process less of a headache and way more fun. Trust me, your future self will thank you.
Thank you, everyone, for returning back for the test session, rather. And I hope you all had a good break. So we're just gonna go straight to the session 2. Basically, we have we're gonna do something a bit different. So, Okechukwu, I think you'll be speaking this as you'll be the second speaker because Ashraf had a lecture, and I think he was thinking previously he was going to speak tomorrow. So the next item on the agenda would be Professor Ashraf El Alali. I'm just going to pull up the Professor Ashraf's so Professor Ashraf El Alali, he's the computational biology and assay professor at Mohammed VI Polytechnic University in Morocco. He's going to talk to us about Africa-based databases to improve agriculture, One Health, and biodiversity. And I basically would let you all know that this is going to be an interesting talk. Ashraf has been doing a lot, especially on the African continent, when it comes to databases, annotations, and it will be an interesting talk. Over to you, Dr. Ashraf. I will make you a co-host just now. You can share your screen. Here, you are now a co-host. Please unmute.
Okay, now I'm unmuted. Thank you, thank God, thank you. And apologies, I had this on my agenda for tomorrow, but unfortunately, just realized it's today. So let me just share my slide as I'm saying this. Okay, can you see my screen?
Yes, please, carry on.
Okay, all right. So thank you for having me. I'm Ashraf El Alali, professor at UM6B, computational biologist, bioinformatician. So today, I'm going to touch a little bit about the different databases and tools that we've developed and how they could reach to this One Health and biodiversity and how it all relates to the mission of AfricaBP. So just a little note about where I'm from. So Mohammed VI Polytechnic University. If you haven't heard of it, it's an international higher education institution established to put research up front and innovation. So we have a vision of driving eco-social economic development through applied research and innovation, not only for Morocco, but for Africa. So this is a big mission. So we are obviously doing our part of, among many other institutions in Africa. So as part of the university, I am the head of the bioinformatics laboratory, which is part of the College of Computing. So the bioinformatics laboratory conducts research in the fields of bioinformatics, computational biology, computational chemistry, and it's meant to serve not only agriculture, environment, but also the health sector. So UM6P, we have state-of-the-art NGS sequencers, we have mass spectrometers, we have also host Africa's fastest high-performance computer and largest high-performance computer. And so our goal at the bioinformatics lab is to leverage the data coming from these NGS sequencers and the available compute to drive the needs of UM6P, the need of Africa to develop innovative research and training programs in bioinformatics and computational biology. So we have four areas of research. We do applied machine learning to different omics, whether it's genomics, metagenomics, different kind of omics, and integration of these omics approaches. We look at genetic markers in plants. So this is kind of where we would be talking today. I don't know if you see a pointer. Let's see if I can use a pointer. It's going to take some time. Here's the pointer. Okay. All right. So the pointer works. So we do GWAS studies, whether it's at the agriculture or even at the medical level, and we do molecular simulations in the drug discovery. So these are four different main research areas. Today we will concentrate on genetic markers and particularly on databases and tools within these approaches. Okay, so we have a plant bioinformatics database accessible via our website. We'll share the link towards the end of the slide or within each tool, and it will be available at the end of the last slide. And so this bioinformatics platform is meant to be both a data hub, hosting data sets that are either computed from local data or that are generated as a result of analyzing existing data sets. We also have several genomic tools, and the idea is to have to reduce the bioinformatics burden from some biologists, agronomes, people who use genomics as a tool rather than being specialists. So we try to alleviate that for several different pipelines and processes. So our platform is accessed, like I said, worldwide. So here is just a quick summary of our statistic page. So we have over 52,000 visitors so far. We've been operating for about almost 2 years, a little bit less than 2 years. We have visitors from these are the top 10, for example. They go from the US, India, Germany. Within Africa, we have Egypt as our biggest visitor. And we look forward to actually serving. We do serve all countries, but this is we have some users from South Africa, from different countries around Africa here. But these are just the numbers for the top users. And then, of course, we have some statistics on how many people actually used our tools, run actual jobs for the last 3 months here, and how many people have downloaded some data from our databases. Okay, so I will go through some of the examples of these tools that we have, and then you can see the full platform on our website. So the first one I want to go over is plant LTR-DB. So transposal elements are genetic elements that can move or transpose from one location to another within a genome of an organism. They are known as transposons, or what, if you've heard the word jumping genes. So there are two main types, DNA transposons that do like a cut and paste, while retrotransposons moves by copy paste. So that's the difference between them, but they are all referred to as transposon elements. They have different, they create genetic variations and drive evolution. especially when they introduce new genes or they alter the gene expression pattern. And we have published some papers showing the importance of transposer elements specifically for plants. So the issue is, they are not easy to, the pipeline to do so is not as easy to find, to actually run on your own, and it requires a lot of steps and further post-processing by in-house code. So we have this database that has all the access, 195 plant species. This number comes from the available, as of 2023, the available plants that have full annotations, within the NCBI. Because we don't just find these LTRs, we classify them, different classes, different clades and things like that, and also how they relate to genes, because that actually changes. They can disrupt gene function, they can cause different variability. So we want to actually give the user information about how they are within the relations to the genes of that particular plant. That's why we only processed here the plants that do have annotation. And then, of course, we classify them. This is kind of the different approaches for LTRs, the different elements that exist. And based on how many elements are present, these LTRs have different classifications. You can get that from our database. For all the elements in the database, you can view them on a map. You can download the data in different formats and things like that. Now, not all of since 2023, there has been a lot of different plants that have been posted. So instead of actually updating the database each time, we have actually opted of making all our code available as a tool for anybody who has sequenced a new plant genome. So since our publication of plant LTR-DB, so now we have a tool called Mega LTR. This tool actually computes, identifies, classifies, intact LTR-RTs for whole genome level, and integrated different tools that are structure-based, homology-based, de novo identification. All of these looks at the different tools and comes up with a consensus among all these different approaches, and then it does classification based on which type of LTR, and then annotation based on how close is it to a gene or not, gene chimeras, and so forth. So a lot of different analysis that one can run. So also we provide statistical analysis, visualizations, insertion time, as you can see here, visualization of these LTRs within the chromosome. So these are actually visualization that we have created ourselves here, and for each genome you get figures. We see our figures showing up in a lot of publications now. So some people take, once they annotate their genome and they run our tool, they take these figures directly into their publications, and we're happy that we started seeing this now. Plant LAI. So LAI is an index that can identify it's an LTR assembly index. It has been used recently as a quality check for genome assembly, particularly for plants. Okay, so the higher the LAI value associated with higher quality of assembly, and there is like so there is a specific range that you can tell that this is a reference genome, or this is a low quality, or this is a gold standard, and so forth. All of that can be found in our publication. So we assessed about 1,644 plant and altered genomes, and we've pre-computed it, because when this LTR LAI came, anything that was submitted before, people don't know how good this plant genome is. They can still access the traditional, scores of quality, but for plants, LTR or LAI value gives a much better understanding on the quality of genome assembly. And so we've computed all of the available genomes at the time that were of good quality. So there are more at that time, but they were so low quality that LAI is not even valid approach. So we've taken all the ones and we've computed it, put it in a database. People can access it, reach the data, ask for, given particular, genome ID, and then they can get the scores. We have some visualizations as well. But again, the same thing we did once we have actually created this tool. Now others will say, well, once we have a new genome, it's not in the database. So as we did with Mega LTR, instead of having to update this database every time, we are actually allowing there is a tool here where somebody could put in their newly annotated, assembled, it doesn't have to be annotated. Once you assemble your plant genome, you can come to this website, you can click on, upload it to our database, and you will get uploaded to our web server, and you will get the LAI score. Now, these are actually all run on high performance computing. These jobs take days to run. So these are not your traditional web services that you will see run in small jobs. So things, go into the Africa's largest CPU, supercomputer. They get computed, and you get that in a few hours instead of a few days. For large genomes, it can take up to 7 days. If you put wheat, for example, it will give it to you, but it will give it to you about 5-6 days later using 56 CPUs. Which is our default value for what we're given here within our service. Okay, so we're not only hosting the data for existing genomes that are already published, we're also allowing new sequencers, new assembled genomes to be able to compute this score as easy as uploading it to our server. We also have comparative genomics. So we do, for chickpea, we have a website that compares these different, not just LTR, but all types of transposer elements within different types of sicer species. So that is also available on our data. It's has the same visualization tools, classifications, all of the things I've mentioned before, but now for all transposal elements, not just LTR RTs. We also looked at, how can these genetic markers be used as in actual plant breeding programs, in actual laboratory work. So we have looked and surveyed the database, the literature, and found, all the people that have used certain transposer elements as markers, and then we made them available in a database. You can search by primer, you can search by type of transposer elements, and you get all the all what you need to do your experiments. Now we also have another tool called Mega SSR. So SSRs are simple sequence repeats. They are also used as genetic markers. This is one of our #1 used tools. This tool gets run, and I want to say 100 times a week minimum. So it's SSRs are one of the easier genetic markers that can be applied in the labs because of the size of the SSRs are not much smaller than LTRs. So you're covering a smaller region, and so they're still the favorite genetic markers. So that's why it's this particular tool is more used than others. But we expect as technology, as NGS gets used more, people will start looking at LTRs as well as genetic markers more as well. So we have a tool that identifies all the SSRs, classified them, allow you to download all the different statistics. You can view them on our website and query the results on our website, or you can download the full output and do whatever analysis you want with it. We have a primer design. So actually this has an in silico primer. So once you've find your SSRs, you can identify which ones you want to actually do in silico primer. And then you can actually check that these, that you will, what kind of bands you would expect to get if you were to run these particular primers in the lab. So it designs the primer for the SSRs that we identify, and also we have a tool to allow it to kind of do in silico PCR validation. Okay, to just get an idea of what you expect, of course. It's not going to replace your actual PCR. Contribution to research, of course, now this is going to accelerate plant species research. This was very, in our workshop, North African workshop, people really, appreciated this tool that were, especially those people who are actually doing plant breeding, needing genetic markers. They were very happy to learn how to use this tool. And again, based on our feedback also on the number of jobs that are run, this remains one of our top tools. We have also a database of for all plants of tRNAs. So tRNAs are non-coding RNAs that are essential for protein translation. So they're important to know. They differ from one species to the other. So we have identified them for all possible plants databases that we could found as of 2022, and they are available as well as all kind of visualizations. You can actually blast against them if you have your own tRNA. You want to see if it exists in which plant. You can blast against our database, or you can download the tRNAs per species, per type of tRNA. So nobody can have to compute this on their own. It's all available in one of our databases. We also have pathways pre-computed for we have over 2.7 million different pathway based for all the available plants as of the publication, which was back in 2021. So it's all available as well here. You for each plant you get all the different pathways. You can actually interact on our maps and select which pathways you want to concentrate on, add new genes and things like that. I'm going to get a little faster because I'm running out of time. We have also a line stats plot. This is a tool that does a lot of visualization and actually computes different multiple sequence alignments, post multiple sequence alignments processes. So once you've aligned your sequences, generating doing different PCA, post MSA analysis, different things that one can do by just giving the your MSA file, or even just the raw data. We do all the steps. We give you all the different clusterings, the trees and the heat maps, PCA, everything gets downloaded by one click. So that kind of alleviates this kind of, because this is a repeated analysis for a lot of different researchers. So we thought we make our tool available as well. So it gets used pretty often. All right, so all of this brings us to what I wanted to really talk about is our vision at AfricaBP is of creating an African DSI data bank, kind of similar to what is available at NCBI or EMBL or the Japanese version as well. So we want to have our own, not similar, not a copy, our own in vision, our own way of doing it as well. So we are looking at having a federated platform, which would be a network connecting African institutions for different DSI storages, analysis, visualization, and sharing. And then, of course, the institution will retain their own data sovereignty while adhering to their own regulations. But we somehow have a federated platform that can allow access to these different data sets that might reside in different databases scattered across different institutions. We're also looking at having this, what's called the global access point. So this would be a decentralized network providing access to sequence data, metadata, and also access to analysis as well. Different analysis, HPCs around Africa that may offer some sort of computational help to different researchers in Africa. So of course, we all know the challenges. We have fragmentation data. Our data is just remaining in different databases. There's a gap of not having enough HPC and computational power for many researchers. And so the impact remains of these data remains, remains unseen unless we actually have this DSI that will actually alleviate some of these issues. Sorry if I'm going fast, but I think I'm gonna run out of time if I don't. So a key component of this African DSI, we want to have a platform, database platform to integrate all the existing different DSAs, as I've mentioned before, and use the federated model to overcome any regulatory challenges. We also want to have a data analysis platform. So this would be a cloud-based or access to HPCs, whatever available, or through bioinformatic tools and web services like the one I host here in my lab. Visualization platform, so allowing people to actually visualize their tools, such as the ones I've mentioned that we host as well, making complex genomic data accessible for analysis and interpretation. And of course, sharing the data, being able to share this among different, among researchers, but being compliant with the national and global regulations. So we want to create this federal database platform by linking existing DSIs, integrate data, by analysis platform, establish data visualization tools, promote responsible data sharing, and engage policymakers. This would be the strategic priorities for us in this effort at AfricaBP. Roadmap. First, we have to build the network, expand the capabilities, and have some long-term goals. I think I will just leave some time for questions. We know that nothing happens without having funding, so we have to, we've laid out some funding mechanisms and government's frameworks. All of this is available on a preprint that you can see, that we have shared at AfricaBP a few months ago. We want to have some, all of this has expected outcomes, enhanced biodiversity conservation, which is one of the main goals of AfricaBP and all our efforts as researchers and genomic researchers in Africa. Strengthen research capacity, so we want to actually increase collaborations among African institutions, improve access to the different tools, different computation among African institutions and collaborations, and of course our support for food security and sustainable agriculture by using genomics in the way it's meant to be used, to improve agricultural resilience and biodiversity conservation efforts. Of course, we call among you to help us engage. Researchers are one of the main stakeholders. If there are any policy holders among us here, institutions, private partners, we call on you to join us with this effort. You can reach, of course, AfricaBP to know how you can help. Okay, thank you very much. This is the website for my lab. You can access the platform through this, and then these are some emails if anybody wants to reach out. And thank you. Sorry, I had to speed up at the end.
Thank you, Ashraf. That's an interesting presentation, and also very interesting to see the number of users. So, I mean, I have a question, but I'm just trying to see if there are speakers who participant on Zoom or on site who have any question. Do you have any participants on site with a question, please? So I can see Hewan Degu. Please ask your question on mute.
Okay, thank you very much for giving me this chance. I am really happy and pleased attending Professor Ashraf's lecture. It is really a wonderful job, specifically working on plant. As you know, we have a lot of indigenous plants and which helps us to secure food. And even if, like, we do have a different diversity, root and tuber crop from the western part of Africa, and also cereal in the northern part, and also, as you know, in Ethiopia also we have a cereal and also multiple crops, including trees and others. And I'm really happy I am learning the great job that you are using. I hope that we are going to access the tools that you have really developed. It will help us because we are going toward this indigenous crop sequencing and transcriptome analysis. Because, as you know, in Ethiopia, one of important resilient crop is insect. And that is where we are using here in the southern part, where in the Sidama region, and specifically here in Awasa. So that will be a great asset for a future collaboration. I think I will communicate with you if you are really willing, Prof.
Welcome.
Okay,
Most
and
welcome.
that is, yeah, so definitely I will disseminate all this information to our university communities, and we'll look for a future collaboration. I'm more than happy to be here. Thank you very much, Prof.
Thank you. The platform is available for all the different annotations. And if you need any other, annotations, any other pipelines, we're welcome to collaborate on those as well.
Thank you. Thank you, Awan, and also Dr. Ashraf. So, is there any other person that have any question on Zoom before we go to participants on site, please? So, while we're trying to see if there are more questions on Zoom, is Professor Elali, some participants are asking if your presentation is going to be available for them.
Sure, I can definitely share it.
Yeah, just share it in the chat box if that's okay.
Sure.
Just feel free.
No problem.
Yeah. Then all right. Okay, participants on the Zoom, please.
Thank you very much.
Ashraf, if you can stop sharing, please.
Stop sharing.
Yeah. Yeah, please go ahead.
Okay, my name is Dr. Emmanuel from Federal University of Massachusetts. I'm really interested in the plants because I'm doing some work using a kind of coconut water to work on import aminoacids. But the question I want to ask is that is there any way we can do the genomics of some of those plants to know the bioactive substance that can bring about curative effects in some ailments like cancer? So these are actually what I was. Thank you very much.
Thank God. Did you hear?
Yeah, so Professor Julian, we didn't quite catch the question. Could you please maybe ask it in a different way?
Let me say what I understood.
Okay, all right.
You're asking if there is any mechanism to help plants become more resilient for something specific? Is that what you're...
What I'm saying is that we can make this where we can actually, when you are doing the ground study, you do some phytochemical and phytohormones to know some substance that are there by active substance that will cause some curative effects medically. And so what I'm asking is that some of these active substances, in any way, since you are doing some plant genomics, gene markers, trying to identify some genes in the plant, is there any way we can identify some genes in the plant that are responsible for the curative effects that those plants contain?
Yes, okay. So that would be the GWAS approach. So we look at different phenotypic information as in response to different traits, genomic traits of or genomic markers of that we have. So that is possible. we can also look at comparative genomics, comparing the different even within for example LTRs, which is what takes a lot of part of my work, looking at different the way chimera genes have certain LTRs that are causing this traits to be available in this plant versus this plant. So there are different known approaches. I just mentioned a couple of them. I don't know if that answers your question. I'm sorry because I didn't hear it.
Yeah, so I think to also add to that, things like the typical annotation work could do, like gene expression as well, right? Where you could find, okay, what genes are responsible for X, Y, Z, and comparative genomics. So there are multiple ways, essentially. The GWAS approach, as you mentioned, there is also the annotation, or even the comparative genomics approach. Where
transcriptomics would be...
transcriptomics as well, right? Gene expression, looking into that.
Or even looking for markers of, if you know this trait happens in a particular species and this could be also possible that it's available in your species, in your plant, you can look at these, use the markers, the SSR markers, LTR markers, to see if they exist also in your particular plant. So that kind of, you could do it PCR without doing sequencing. So people do this. This is where the genetic markers are very useful because they are cheaper way of doing this without sequencing the full genome.
Yeah. Thank you, Ashraf. Professor Julian, do we have any more questions on site, please?
No question here. So we might proceed to make progress from there on. Thanks.
Thank you so much. Thank you, Ashraf. Please don't forget to...
No other questions.
Yeah, don't forget to put your presentation in the slides in the chat box so people can have
sure.
Yeah,
will do.
All right, so we're going to go to the next presenter, which is Okechukwu. I am going to, because I'm supposed to share it from here, so I'm going to find her slide. Do we have Okechukwu on the... Okay, I am going to make her a co-host. So Okechukwu, I've now made you the co-host. Please unmute yourself, and I'm going to find your slide. All right. Okay, here is your slide. And I'm going to share the screen. So can you see my screen, please? Can you see my screen? So can you see my screen, please?
Yes, we can see.
Okay, all right. Chikoli Okechukwu, could you please go ahead and present? You're already a co-host. Could you please?
Yes, I can.
Okay, all right, go ahead. Go ahead and tell me when you want me to move the slide. Just let me know so I can move it. I can go to the next slide. Hello?
Hello.
Yeah, please go ahead and present, please.
Okay, I'm presenting on the cytotaxonomic analysis of 3 Annona species. The deciduous plant apple family species.
Should I go to the next slide?
Made up of about 125 species. It has edible fruits and of medicinal values too.
Okay, I think we can't have. Looks like Kachiko is having issues in terms of the internet. Because the internet is unstable. Yeah, I'm gonna have to stop the presentation because we can't. Yeah, so we probably have to go to the next presenter. So do we have Dr. Mustafa Ayo Pupola on the call, please? Professor Julian, do you have Dr. Mustafa on site?
Yeah, no, he's not there.
I'm right here. I don't know why. It's network issue.
He's not here.
Okay, so we're going to move to, let's see if we have other speakers that are on site that are going to. So I think we have Ansia on the call now. So Dr. Ansia, would you be able to speak now? Vancola? I will make you the co-host while we kind of just keep the other speakers and then return back to them. Ansia, you have a co-host. Could you please let me know if you can hear me?
Hi, thank God. Yes,
Okay, all right.
Shall I
Thank you,
present?
Ansia. Yeah.
Yes, I can hear you.
All right. So Dr. Ansia will talk about, so no, she's supposed to speak at 4:10, but I think we need to keep Dr. Mustafa to see if it's on site or online before returning back to them. So Dr. Ansia will talk about the genome assembly of the indigenous African plant species, Helichrysum. Dr. Ansia, are you ready to speak? All right. Okay, here you go. I am gonna just spotlight you. I hope I can do this. So I think I can spotlight you. All right. I can't spotlight you unless you start your video. So you want to start, you want to put it on so people, that way I can spotlight you. Is that okay, Ansia? You're on mute, Ansia, if you're talking.
Sorry,
Ansia.
I'm just getting my stuff ready.
All right.
I was ready to present right now. Let me just switch my lights on. Is it all working now?
Yes, we can see. Let me see if I found you again in the. So, okay, I need to spotlight you. All right. Okay. Yeah. I think you're now part of the spotlight. Yeah. Perfect. So share your screen.
Sorry, my stuff is just jumping around. Okay. So hi everyone, I'm Antia van Koller. I am a senior scientist and bioinformatician from the South African Medical Research Council's genomics platform. And today I'll tell you a bit about the genome assembly that I've been working on for a complex African plant known as Helichrysum odoratusum. So the The name of this species is based from the Greek word helichrison, and this is because of the golden yellow flowers that is characteristic of this genus. And the word odoratissimum refers to strong odor. That's because this is quite a fragrant plant, and that's why it's also known commonly as African incense. And in our native languages, it also goes by koichut or impepo. So this plant grows quite abundantly throughout Southern Africa, and it is one of the most popular traditional remedies. It grows abundantly everywhere, in the highlands, in the little slopes next to the road, not just in South Africa, but also in Mozambique, Zimbabwe, Malawi, and a little bit more north as well. It is of great cultural significance in South Africa. So parts of the plant is routinely burnt to rebuke evil spirits, or it can be inhaled for good luck or good fortune. It also has lots of medicinal uses. It's been used for the treatment of heart disease, chest pain. It has calming effects. It's also been successfully used to treat cough, fevers, urinary tract infections, which suggests that this plant might have some antimicrobial properties as well. And again, the calming effects of the plant has also been used to treat things like psychosis, and this could be due to the presence of phytocannabinoids, which has been identified in other Helichrysum species. It's also used commonly as an insect repellent or perfume because of the strong odor, and it is one of the most frequently harvested and traded plants in South Africa, and it has a great potential for commercialization. So unfortunately, no fully assembled genome currently exists for any of the Helichrysum species, but there have been some studies on the karyotype and the genome sizes. And these studies show a lot of variation in genome size, chromosome number, and ploidy level. Multiple different levels of ploidy have also been reported within a single species, suggesting that this is an important evolutionary mechanism for this genus to drive diversification and ecological adaptability. So because there's no nuclear genome assembly available for any of these Helichrysum species, we then sought to sequence and assemble the genome of Helichrysum odoratissimum. And today I'll be presenting those results to you. So just a little background, because I'm sure not all of you are working with genome assemblies every day. So obviously a reference genome is very helpful for enhancing genomic research. But if there is no reference available, we need to construct it in a de novo manner. So after sequencing, this process relies on alignment tools that can align all the reads to each other in order to identify overlaps to make contiguous sequences referred to as contigs. And the overall goal is just to create one long contiguous sequence that encompasses the entire genome, but that's not usually how it goes. So you're more likely to end up with little fragmented bits, and you can do the process of scaffolding, which allows you to pull individual contigs together. But yeah, overall, this can be a quite computationally intensive process, and things like long read is really helpful for long read sequencing is very helpful for de novo assembly because it's a bit easier to resolve repetitive regions and inverted regions, which might struggle if you only have short read data. So we got a fresh plant and we were able to extract high quality DNA and RNA. And then we performed ONT sequencing on our Promethion P2 solo sequencer, and we did the high-C and RNA sequencing on the MGI-TNBC G400. These are just my colleagues that were working on this. So this is the overall plan for the assembly. We used the ONT data and I did the super accurate base calling to ensure that we got the best quality of data out. And this was followed by QC using nanoplot, trimming of adapters and filtering based on read quality. And we also used the ONT data to try to estimate the ploidy and the genome size of the of this individual. For the Hi-C data, we also did QC and of course the basic trimming and quality filter to make sure that we're only working with the best data. And I then used Hi-Fiism to do a de novo genome assembly, which integrates both ONT and the Hi-C reads. And the Hi-Fiism, the newest version, works really well with ONT data. I also took the error-corrected ONT reads to redo the ploidy and genome size estimates, just because it would be a bit more accurate with the error-corrected reads. After I had the draft genome, I integrated the Hi-C data again by doing alignments and lots of pre-processing to find the Hi-C contacts that would be informative of the 3D structure of the genome. And I used that to do the scaffolding using YARS. This was followed by lots of assembly QC and manual curation of the genome to ensure that everything is in the correct order. And then afterwards, we will do annotation. So some studies have been done on this exact species, Helichrysum odoratusum, and it showed that there were both diploid and hexaploid individuals within the same species, and this was dependent on the location where the plants were. So smudge plot analysis showed that this individual is hexaploid, and the genome scope analysis agreed with this, and you can see the 6 peaks with the KMA coverage. This also revealed to us that this genome is highly heterozygous, so have about 5% heterozygosity, which is quite high. I think anything above 1% would be considered slightly more complex than normal. And so, yeah, and it was quite a large genome size with up to 750 megabase per, megabase per haploid size. So this is the Hi-Fiism command that I used, and of course referred to the ONT flag to indicate that this is ONT data and integrated the Hi-C data. I specified that there are 6 haplotypes, and that's why it produced assemblies for the 6 different haplotypes. Obviously, the higher the ploidy level, the less accurately Hi-Fiism can split the haplotypes, but it does improve the quality of the primary assembly. So for my further downstream analysis, I looked mainly at the primary assembly that was generated, which is essentially a collapsed version of all the haplotypes. And then I also looked at haplotype 1. So you can see there's a lot of contigs in this, so it is still quite a fragmented assembly. And there's some difference in the total size between the primary assembly and the hap1. But overall, not a bad assembly if we look at the N50 and all the other metrics. So due to the very large number of small contigs that we found in these assemblies that we couldn't integrate elsewhere following scaffolding, so I did a lot of trial and error. We decided to just extract all the contigs that were smaller than 50 kb, and then we compared these sequences to known mitochondrial and fluoropath databases. And then if more than 50% of a contig matched to these databases, we would remove that contig prior to scaffolding. So here I just show for the primary assembly and the filtered assembly, I removed, you can see quite a lot of smaller scaffolds were discarded, and it didn't affect the total length much or the completeness of the genome. So in the bottom here, I also have the BASCO score. So these are pretty complete assemblies, although there is quite a bit of duplication, but this could just be a normal observation for this species. So we integrated the Hi-C data. Now I think tomorrow there will be someone presenting on the details of Hi-C library prep and how this works, but just for a quick summary for those of you maybe not familiar. So Hi-C is a high throughput technique that is used to capture 3D chromatin conformation. So we know inside the cells, the DNA is folded in on itself, and through a process of cross-linking, we can ligate ends of the DNAs together that are in close proximity. So you can end up with DNA fragments that are not necessarily linear as they should be, but rather contain information from different pieces of DNA that are in close proximity to each other. And then once you have these fragments, you can prep and sequence the library as you would any other DNA library. And you can then generate a contact map like this, where you can see which regions of the genome interacts with the other regions. So our Hi-C data was really good quality, and from the pre-analysis that we did, or the pre-processing, I could see a really good capturing of chromosomal interactions with really good resolutions, with detecting differences at various interactions at various different distances. So this is the assembly metrics before and after scaffolding for both the primary and the haplotype 1. And you can see that the contigs did reduce in both cases, and you can see that the N50 significantly increased. My favorite part of these tables to always look at would be the L90, which in essence is the number of contigs that make up 90% of the genome, and you can see that has been reduced to 24 in the primary and to 34 in the HAP1. So a majority of the genome is made up by a small number of contigs, which is a good sign. So the next step is then the genome curation process, just to make sure everything is in the right order, and then we can assign the chromosomes. So the process of doing this, you would take the scaffolded assembly and generate a TPF file, which dictates the order of the assembly. And then you would also realign the Hi-C reads to the assembly in order to generate that contact map, which you will then manually rearrange and assign chromosomes. And then you combine all that information. In the end, you would end up with your curated assembly. So how the manual curation process works, sorry, this is not a great picture, but I think it's very useful. So when you generate the Hi-C contact map, you can see how different regions of the genome interact with each other. And you can see, for instance, in this example, if there's a little spot here, it indicates that that region of the genome likely belongs over here. So the manual curation process is basically just manual splitting of scaffolds and rearranging it until it's as meant to be. And here is an actual image of my Hi-C contact map for haplotype 1. And you can see that there are some interactions here off to the side, which we can then manually incorporate these scaffolds into these bigger scaffolds. And even if I zoom in on one of these scaffolds, you can see even within a scaffold, there is some rearrangement that needs to happen. But of course, this is a very complicated plant. It's hexaploid and it has a lot of repetitive regions, so it does make this process a bit more difficult, but we are gaining some momentum there. So yeah, our next step is just to finish off the manual curation of the genome by rearranging the Hi-C contact map on a pretext map. And then we can define the chromosomes, and we can then export the data to generate the final assembly, which we'll upload to NCBI. And then we will do some genome annotation using the final assembly and the RNA-seq data that we have, so we can finally see, you know, what is it about this plant that makes it so interesting. So yeah, just like to acknowledge everyone at the SAMRC and of course everyone at AfricaBP that have been working with us on this. And yeah, thank you for the opportunity to present here, and thank you all for your attention. Does anyone have any questions?
Okay, thank you, Ansia, for your presentation. Do we have any more any question on the chat box or on the Zoom call before we move to the on-site participants? So someone is saying, Mary Emaragi. Mary, do you want to unmute? So Mary is asking, I'm just going to read out her chat here. Let me see. So she's asking why chromatin rearrangements. Not quite sure the context of. Mary, do you want to provide more context to your question? So I'm just gonna. I think you should have. You should be able to unmute now and ask your question.
Thank you. Can you hear me?
Yes, go ahead. We can hear you.
Yeah. I know that I have not much knowledge about genome assemblies, but chromatin, like it looks to me like, are you trying to associate parts of the genome to see which ones are related? And what was that? What do that do to assembling genome? What are you doing? What's the strategy here? Thank you.
So if you're only using DNA sequencing, I think nowadays the assembly algorithms are very good where you can get a telomere to telomere assembly using just, for instance, very long ONT or PacBio reads. But for some of these species, it's a bit more difficult. When you just use the whole genome sequencing, you end up with very fragmented genome assembly. So because the assembler is trying to, build the puzzle to put the assembly in order, but it doesn't necessarily know which order it is in. So if you combine the whole genome sequencing data with the Hi-C data, which captures the chromatin conformation, you can associate the different contigs with each other based on their spatial proximity. So because the Hi-C process ligates pieces of DNA together that are, we assume, within the same chromosome, and the closer they are to each other in the chromosome, the more intense the interactions will be. So based on the chromatin conformation, we can generate those contact maps to see exactly where the genome is, like the exact order of the chromosomes, or at least to see which chromosomes go together. And then we can define them based on their lengths. I hope that answers your question.
Thank you, Ansia. So I hope that answers Mary's question. Hewan Degu, please, you can unmute.
Thank you very much, Doc, giving me the chance. It is a wonderful presentation. I learned a lot because it's new for me, the plant. I think it is African indigenous plant. Like one of my questions is like why it is really selected. Is there some kind of different variation or medical aspects? If you just briefly explain for me that. And then when I just wanted to know, because there is a number of ploidy in African plants. So have you ever seen some kind of repeated elements in the genome? And is there any ploidy events observed in this genome? And that is some of my question, but now I will read your papers if it is published, and I will look for more exposure in the future. Thank you.
Thank you. So this species was selected because it is of great cultural significance in South Africa. It is one of the most commonly used plants for traditional medicine as well, and it is expected that it likely has some compounds that are antimicrobial and have calming effects, maybe like the phytocannabinoids, which can have good effects with like inflammation, etc. So yeah, we are hoping to, annotate the genome to see if we can find the molecules responsible for this medicinal effects that the plant has. And in terms of ploidy, yes, so it is known even within the species that there are different ploidy levels based on the area in which the plant grows. And we do suspect that this could be due to just like a whole genome duplication that has occurred. So in that case, you would see a lot of duplicated regions of the genome, and this is something that does commonly occur in these plants with their large genomes. It's not this is not published yet, but we are hoping to publish this soon.
So thank you, Ansia. We have another question from. Looks like you have a lot of questions, Ansia. We have one question from Laide Rasaki. Laide, do you want to ask your question orally, or shall I read it out over here? So he's asking, I am curious to know how you name the chromosomes if there is no reference genome already. So I think this is, yeah, there's a context already. Ansia, you can answer this one.
So we would name the chromosome based on the high-C contact map. We would be able to see which regions interact most with each other, and so based on that, you can define the chromosome. But there's also some previous studies where they do like flow cytometry and stuff where they can estimate the number of chromosomes that are present. So we'll then see if what we can identify based on the high-C contact map and how that corresponds to the information that is known about like the number of chromosomes, for instance.
Okay, thank you, Ansia. So I think we're trying to go back to the on-site participants. There are some questions there. So they lost connection, so I am trying to get them back up. All right, Julian, I am going to make you a co-host. So Professor Julian, do you want to start your video? And then are there any questions on site, please?
Yes.
So do we have any questions?
Thank you very much for this nice and insightful presentation. You made mention along your presentation that this plant. Yes, we have a question here.
Please go ahead on site. I'm just seeing the control video now.
Presentation that this plant has an insect repellent insect. Am I clear?
Yeah, go ahead. Please carry on.
On mosquito. So I'm thinking of a way that this gene is responsible for producing strong odor is amplified. Meanwhile, the enzymes strong odor are amplified such that we can make it
So I think we could barely hear them. Ansia, are you hearing anything?
In much quantity, so that it can be used for commercial activities and to reduce.
So we could barely hear the control. Could you please type
Okay,
your...
let me repeat the question. I said the...
Yeah, could you please type your question, write down your question and send it to the IT or the control, and then they'll paste it in the chat box so we can
Let me
ask it.
make the question clear again.
Okay, all right. Go ahead. Okay, go ahead. We can hear you now. We can see you rather. So we're trying to see if we can fetch the question from the on-site participants. Control, do we have the question now? Do you want to paste it in the chat box so we can read it out to answer?
An insect-dependent ability. Were you able to identify that genus? Done that. Can you share with us interest and your contribution? That is, I want to have an input, your input personally, on how we can amplify that gene so that it will be of a commercial interest, or it can be used to produce. It can be used to produce something that will repel insects, that mosquitoes particularly, because that is where I have an interest.
Okay, answer. Did you get his question? He's talking about the translation. Yeah, I think we got your question, though it wasn't so clear, but it's fine. So he's talking about how do you get a gene and then transform it, and then translate it to an actual thing that you can use. So if you can just, within the context of what you heard.
Yeah, sorry, I didn't hear the full question, but I think it was relating to how would you identify which genes may be contributing to specific effects and how that could potentially be commercialized. So this would be hopefully identified during the annotation process, because we also have RNA sequencing data. So we can use the genome and the RNA data to try and identify all of the genes that are being expressed, and then we can compare that based on homology to other plants to see if we can identify things that have been expressed in other plants that have had, medicinal effects, etc., and see how ours maybe differs from that. But yeah, it's most likely going to lead to additional studies to see if those genes that we'd identified are the ones, contributing to the effect that we're trying to look at. And then if it is established that there is a link between specific genes and a specific molecule that is produced for whatever effect, whether it's, insect repellent or something else medicinal, then that could potentially go into the commercial space where they can try to make that into a different project. But that's potentially not something that I myself will be doing, because that's not my field of expertise. So I'll leave that to the biotech experts.
Yeah, thank you, Dr. Ansia Vancola. Do we have any more question on site, please? Question on site? Any more question? Ansia, it looks like you've got lots of comments. Question. Sorry, do you have any more question on site, please?
No question.
All right. All right, thank you so much. We have another question here. I could get lots of feedback for you, Ansia. It looks like, yeah. So Matthew Moses says, please, my question is more like a recommendation. I'm a graduate student of medicine and surgery. I'm keen. Okay, I think kindly highlight what I need to have or know to succeed in this field. So I don't know if that relates to you, Ansia, or not, but you can just, this looks like more like career kind of stuff. So over to you.
I mean, I don't know if I have all the right answers for that one, but yeah, just see if you can get into a program where you can do research. I think you're in a good space at the moment. You can see who's working on what, and if there's anything that you find interesting, just reach out to those people and yeah, just put your passion out there and make it happen. Good luck. And yeah, thank you. Thank God for the opportunity to present here today.
Yeah, thank you, Ansia, and thank you for coming much earlier to present 1 hour earlier so that we can be able to align with other people who are not here. Hopefully they come in much later. And yeah, and hopefully we'll see you again in during the East and Central Africa workshop to talk about the genome. Thank you so much. All right, perfect. We're gonna go to the next presenter. Again, he was supposed to speak in the next session, but we have to bring him forward. That would be Michael Landy, Kofi Landy. Michael, if I'm just gonna make you the co-host and please unmute yourself. Michael is at the International Institute of Tropical Agriculture in Kenya and is also a PhD student at SLU, Sweden. He'll be talking about the genome assembly of the white star apple as part of the African BioGenome Project. So, Michael, do you want to unmute yourself, please?
Yes. Can you hear me okay?
Yes, please, we can hear you.
Perfect. Hi, everyone. My name is Michael. Just allow me to put off the video just in case I experience any internet connectivity. So we'll start by sharing my screen. Okay. Can you see my screen now?
Yes, please, we can see your screen.
The whole, okay, perfect. So let me
Yes,
just
carry on. Yeah, carry on.
All right, so as thank God mentioned, I'm based in Nairobi, Kenya, and today I'll talk about the African star apple genome. But for this talk, I will not talk about the introduction of this crop. I will dive into the genomics. We will have, I think, tomorrow 2 talks on library preparation and also propagation of this plant, this fruit. But as you can see, it looks a really delicious plant, and I'm really looking forward to visit West Africa, Nigeria, to test this fruit, the star apple fruit. When we think about plant genomes, we think about stories that we either talk to our children, and when you read a story from a book, that will be probably the genome, and the pages would be sort of the chromosomes, and the pages that you read will have the instructions that probably have you think in the genome world will be the genes that sort of give us the instruction, the functionalities of our plant, and why we care about plant genomes is these are just examples of the applications of plant genomes, and just to mention a few, if probably you want to think about crop improvement and breeding, sort of looking at wild type plants and maybe transforming them to domesticated plants. Obviously, plant genomes come in handy if you're thinking about resource conservation and ecological resources. I think this is probably the main theme of this workshop. So genomes come really in handy. If you're thinking about plant evolution and classification, genomes also are really, really important. And just to mention about epigenetics, thinking about beyond genetics, then you have to also understand the genome to explain any traits that are influenced by an epigenetics, or rather the environment. So in the world of genome assembly, I think probably Dacian already mentioned, but think of it as solving a jigsaw puzzle where you have little pieces that you try to join together to have a bigger picture, and that is the genome. And just to give an overview of genome assembly, is these small pieces in a jig puzzle would be your short reads, short or long reads. So small pieces of DNA. I'll explain how they're produced in the coming slides. And as you're solving this puzzle, you tend to join these pieces, and these reads, you join them to form contigs, sort of bigger, smaller chunks of these small pieces. And these contigs you can also assemble them to scaffolds, and later on you have a full representation, for example, a chromosome, and within a genome, say, for example, with the staple genome, you'd have all the chromosome, and then you can define actually the functionality of a plant from a plant genome. But while we always do this the process of genome assembly, there are always some problems that we encounter while assembling these plant genomes. On this plot here, what you can see is, if you imagine this a plant is a diploid inbreed species, where you expect two copies of the genomes coming from the two parents are homozygous, then it's always easier to represent this genome as single haplotype reference assembly. And by haplotype, I mean one copy of a diploid. So a diploid will have two copies. So one copy will be the haplotype 1 and haplotype 2. And for similar copies, sort of homozygous genomes, you only need to have a single haplotype reference. And this is pretty straightforward. But when we have cases where we have outbred assembly, then it becomes a bit tricky. And you have, so here in gold and blue, these are contigs, or sort of a chromosome coming from one parent, so the maternal and the paternal. And always when you're assembling these pieces, you sort of don't know where the pieces are coming, either from the father or the mother. And you can always represent this either as a collapse representation, and how we call this we call it as haplotype mosaic reference, where you represent this as combined if you cannot really differentiate which contig is coming from which parent. But in the recent times, we do have some well-refined sequencing platform that I'll mention in the coming slides, where you can really define where contigs or scaffolds come from. And then you get a haplotype first reference assembly, where you know that, okay, this haplotype 1 is coming from this parent, and this other one is coming from the other parent. And this is specific for diploid, an example of diploid genomes. And so, as I mentioned, the long reads sequences, and here I specifically mentioned the Hi-Fi reads that are making us plant genome assembly to be really easy. And the fact that, and I will not get to the details of how this process goes on. I think we'd have a presentation on library preparation and get to understand it more. But just to point out here, the Hi-Fi reads have very high accuracy, an accuracy of more than 99% accuracy. And with this accuracy, you have the advantage of accuracy, and also having long reads. And if you have these two combination, then it's really easy now to assemble a genome and to have really high quality genome assembly. So just to jump in on the workflow of the genome assembly of this species star apple, and just to mention here in green, these are the steps that I will share. So once we have this Hi-Fi reads, we do the quality control, because this organism has not yet been sequenced and assembled. It is always nice to estimate the size of the genome and also know the ploidy level, how many copies of this genome in these plants. And I will share some of the assembly statistics after doing assembly using this tool called Hi-Fi SM, that I will share the different steps. And then check for duplications and remove if there's any duplication rates within the genome, scaffolding. And in the end, in red here, these are steps that we are ongoing. And I need to mention that this work is ongoing. So some of the results are preliminary results that I will share. And so after scaffolding, normally what you want to do is you want to do manual curation. You want to gap fill and remove any contaminations that are present in the genome. Evaluate the assembly, and then obviously at the end you want to look at the gene repeat and the repeat annotations within the genome, so that you can explain some of the functionalities of this plant. And so the data that we use in this case is the PacBio Hi-Fi reads that I mentioned are really good quality. And we sort of use additional information, that is the OmniC. These are the reads or data that we use to do the scaffolding part of the analysis. As I mentioned, the quality is really good. And here I'm showing, so the first plot is Hi-Fi reads, and in the bottom is the OmniC reads. And here is just the length of distribution of your library. And what is interesting to note, we always have reads that are way too short and reads that are way too high. And the first thing what we need to do is to filter out those extreme lengths. And what we did is we filtered the Hi-Fi reads based on the length of 10 kilobase pairs and to 30 kilobase pairs. And for the OmniC reads, because these are short reads, and so we only filtered any read that is 50 base pairs and above, and that's what we kept. And here I don't show, but we filtered out some bases upstream, so 5' upstream, around 15 base pairs. And for the OmniC also, we trimmed 14 base pairs upstream that were a lot of biases. And what I mean by biases is we couldn't, from the quality control, you can always see unproportionality of the bases within your library. And so we ended up with a total number of reads for the Hi-Fi reads, 3 million, and the length, as I mentioned, 10 to 30 KB. And then this was the also total length of the reads for the OmniC that we take to the downstream analysis. Now, for the first step is to, once we have our data, our clean data, what we want to try to answer is what then is the estimated genome size and what is the ploidy prediction. So what you see here is a plot from a tool called GenomeScope. And here, what you're seeing, these are KMA plotted. So this approach will try to estimate the size, and here it tells you the estimated size of this genome in one copy is around 822 megabase pairs. And you could already see that is a very highly homozygous genome of 99.7% and very low heterozygous level of 0.2 3%. And already here you see like the first peak will be your heterozygous KMAs that are pretty low from the percentage. And the second peak are homozygous KMAs that shows this genome is highly homozygous. And we used other tools called SpargePlot to just predict the ploidy prediction. And as you can see, so we use different types of KMA sizes to do the prediction. And as you can see towards the end here, AB, it estimates 82% that this genome is a diploid genome. And if we also do another trial with 31 KMA, then we get 78%, which sort of these two approaches tend to confirm that this is a diploid species, and the estimated haploid size is 822 megabase pairs, and it's really a highly homozygous genome. So after this prediction, we went ahead and do the genome assembly, and here I'm just showing the method Hi-Fi SM. We used additional 2 other methods to assemble these reads, and I will share the statistics, but just to focus on Hi-Fi SM because this was the main output that we take for the assembly. And what happens here is you have your input Hi-Fi reads, and as I mentioned, you'd have a set of reads coming from here in orange is from one haplotype, and then here in blue is from the other haplotype. And so this region here is an example of an heterozygous variant. And in the green, these are homozygous reads, so reads that are present in both haplotypes. And so what the tool does is first it will check for any sequence error and correct this, and will identify sites that are actually the heterozygous alleles. As you can see here, there's AAA, and from the other haplotype is CCC, which is the different allele. And so after it corrects, it sort of starts to build a graph of this genome, and these bubbles here are where we define reads coming from different haplotypes. And so once you get to these graphs, you can either go two ways. Either you get the primary assembly construction where you only have the primary with one of the variant and have sort of some alternative assembly, or if you have other complementary data, and these are actually reads coming from both parents that help you to separate these two faces, which we didn't have for our case. And so we ended up representing this genome as a primary representation. And also the fact that it's highly homozygous, then getting the primary assembly of the genome was an ideal approach. And so after doing the assembly, we now did some statistics, and what you can see here is a screenshot of the statistics of the genome. So the first row is outputs, the primary assembly of Hi-Fi SM, and HiCanu, which is another tool that we use to assemble this genome, and File is another tool that we use. But just to draw attention on 3 key characteristics. So first is the number of contigs that the assembly generates. So you can already see around 7,111 contigs are only for the Hi-Fi reads. So what you'd expect is you want a lesser number of contigs. So the HiCanu and Fly tend to have a lot more contigs of the assembly. And in terms of the total length, well, this was really surprising because we already saw the estimated genome was around 820 megabase pairs. But here we are seeing a huge number of total length. So this is 2.4 gigabytes. Even more for Haikanu and Fly. Fly I got 15 gigabytes, and this was 2.8 gigabytes, which is really, really huge. And also another factor to check is the N50 value, which was really good for Hi-Fi SM, 1.3 megabase pair. So this tells you the contiguity of the assembly. And for the Haikanu and Fly had very low, and this sort of already showed that these two assemblies here were highly fragmented. And that's why we move forward with Hi-Fi SM assembly. So then with this primary assembly, we went ahead and checked for the completeness of the genome. And tools like Busco will tell you how complete this genome is. And you can see here it's 98% complete, but then the duplication rate is really high, 87.6%. So this was really puzzling for this genome. So what we do next is we look for regions that are highly duplicated. And what we use is we use a tool called PatchTubes that has two main steps, where the first part, what it does is it takes your long reads and then maps it to the reference that did primary assembly. And what you get is sort of a coverage output. And then the next thing, what it does, it splits the assembly into chunks of say 10,000 base pairs, for example. So split assembly by a certain number of length, and then it sort of does a self-alignment, right? So using the self-alignment and the coverage results, it checks for haplotypes, or rather contigs that are identical, and filters this as exact matches. And so it removes this duplicate this way, and you'll end up having a patched genome and some of the contigs that were actually removed. And what is interesting with this tool is you can redo these 3 steps until you get a decent haplotype set, haplotype set that is either less duplicated, and it's also close to your estimated value. So we did this step, and then we moved forward and now get the statistics of the draft assembly. And here is the same that I mentioned before. So we have the number of contigs. So the first column is the primary assembly, and then the first round of paging, so the page gap, and then the second round of paging. And you can already see in the total length from 2.4 gigabytes, then we get to a close estimate of the genome, which is around 821. And in terms of the N50 value, the last column, as you can see, is 3.1 million, which is really improved contiguity as compared to what we had in the draft genome. If you look at the duplication level, so for the second round of paging, we have a 6.6% duplication rate, which is actually more expected if you compare with even other genomes that have already been assembled. So this is really good. So we move the next step, where we do the scaffolding using now the OmniC reads. And for the statistics again, for the star upward scaffolds, we now have 596 number of scaffolds. The length is 821 megabase pairs, which is really what is close to the expected size. The N50 is 57 million, which is highly contiguous, and this was good. And already with the L90 value as 13, this sort of gives you an idea of the number of chromosomes that you'd expect for this species, and normally it's always +1 or -2, +1 or -1. And just here to show is a contact map showing the scaffolding of this genome. And the next step, what we do is then to do the manual curation of this genome. So once we've done the scaffold, so we need to manually curate this genome. And then because we have the scaffold, so we expect to have gaps that we need to fill and check for contamination within the genome. And we will submit this genome to NCBI and later on do the genome gene annotation and just to have an idea of the repeat content of this genome. And a point that I didn't mention is having the write-up and submitting it to be accessible to everybody. And just to conclude, so we expect to achieve a very high quality of star apple genome, and this is the best, the fact that we already have high quality reads both on the OmniC and the Hi-Fi reads, and this would actually be relevant in the genome-assisted cultivar breeding to provide a better understanding of important metabolic pathway. And we will actually see some of the benefits of this crop in the coming days from the other presentations. And we would also use this data to do some capacity development, where we will do some training workshop on genome assembly and annotation to build regional capacity and promote the in-continental transfer of knowledge. And now I'd like to acknowledge everybody who was involved in this project, the INCABA who did the sequencing, and we will hear about them in the coming session. African BioGenome Project, led by Thank God, for actually organizing the whole workshop and also managing the whole analysis. IITA, where I'm based with my supervisor Andreas, and the team from ANCAB and IITA who have really assisted in making this a success. So thank you very much, and I'm happy to take any questions.
Wow, thank you, Michael. This is this is very detailed and good to see the progress. So we're just gonna, if you could please stop sharing your screen, Michael, so we can have the two screens together. We're gonna take questions. So do we know if people on site, do we still have people on site? Control? Is Control still on site? Are they still online, or just trying to see? Oh, it looks like we lost the on-site people, the connection. So we're going to take questions from those on Zoom, the participants on Zoom rather. If you do have any question, please use the icon to raise your hand or put that in the chat box, please. So we have some questions here. So Hawan, do you want to ask your question orally to Michael, please? Hawan, let me try and find you. Yeah, Hawan, you can unmute now and speak, please.
Okay, thank you very much. I was watching and listening and learning a lot, specifically this star genome plant is really has a reference genome. It's because this is my childhood fruit, which I only know eastern part of Ethiopia. And it is really great resilience, and the wood is really great. But no, I didn't find that, no research at all. And recently we are collecting this germplasm to disseminate to the northern part of Ethiopia, because as you can see, in the northern part there is a lot of degradation, and the forest is shrinking. But this is a fruit and by itself, and the wood is really great. And I'm so much happy to find out, lastly, the Africans are really sequencing this genome. So my question is, when you are doing the reference genome, as you know, the distribution of this plant is in the equator, I think around that, and a little bit to the north and a little bit to the southern part of Ethiopia. No, to the southern part of Africa. So when you are doing the reference genome, which accession or a variety have you used? And is it like, a kind of, can we trace the plant passport data? That will be really interesting. Then it will help us to proceed with our research in this plan too. Thank you very much.
Okay, that's really nice to know that the fruit is you grew up taking the fruit. That's really nice. So onto your question, I'm actually really looking forward to actually test the fruit itself. Yeah, so to your question, so I think the accession was from Nigeria, but I think we will get more of the presentation on propagation of this plant tomorrow. Yeah, so maybe then we can have I think the accurate accession of this plant. I don't know if thank God you know, because I have no idea.
Yeah. So, Hewan, this is from the IITA collection, the accession, and the presentation on the wet lab and the sample collection and processing part of this work will be presented tomorrow by the IITA scientists and by the genome facility where it was done. So you will get more background information on that. But definitely, it's the collection at the IITA. It's been there for a while, and then yeah.
Thank you.
Okay. Yeah, it's good to see that you like the fruit as well, right? I'm sure many people on the call. So for many people who perhaps cannot map the common name to the English name, also this is agwalumo or udara, for instance. For many people who know the local names of the species, so it's hawked everywhere on the streets in West Africa. And as you just rightly also pointed out, Hawaii is also found in Ethiopia, which is good. Okay. Thank you so much, Michael. We have an additional question. Professor Anemugai has a question, and you want to go ahead, please?
Yeah, I think for me, I just want to appreciate the two scientists because it's showcasing the fact that we can do this work on the continent. My question to both, very briefly, you've been able to present very high quality data. It'll be interesting to hear about the start and whether it was as easy as you have projected it to be. What are the challenges, for instance? Just off the cuff, what are some of the challenges you faced? Was it the sampling? Was it the isolation of the nucleic acid, or what specific challenge did you have? Thank you.
Maybe we can also spotlight Ansia here. So it looks like this is a question for both of them. So Ansia, if you want to put on your video or something, so I can spotlight you if your video is off. So I can do it and then you can go take it back as well. So you want to put on your video, Ansia? So that I can spotlight you and yeah, okay, found you. All right. So this is more of a question. There's an important question, Professor Mugai, on what it is. So I'm sure you both heard about it. So you want to just talk about it, and I'm sure that would also help inspire others. Because one of the things AfricaBP aims to do is to sequence the genome locally. No matter the challenges, no matter anything, it has to be local. And of course, many people already have seen that. So let us know in the next 5-10 minutes. I want us to be very detailed, explanatory, and yeah, just say over to you.
And do you wanna start, or I can?
Sure. Okay.
Yep.
So some of the challenges we face. So I am not a plant bioinformatician. I work mostly with humans and animals and pathogens, so it was very new for me to work on plants. But AfricaBP has been really great in, linking me with all the right people, because you don't know what you don't know until you realize that there's more to know. And another challenge that we specifically faced was computing resources. So because the plant that I was working with was so complex. A lot of the typical resources that we throw at assemblies wasn't enough to resolve this complex genome. So I'd had to go, just speak to other African researchers and to see how they do things and how I could improve how I was doing it. And yeah, we've made some good connections with thank God's team as well. I think the Sadiq has been very helpful, and I've learned a lot through the whole process. I'm just very grateful and happy that we can sequence African plants on African soil and analyze it ourselves. Over to you, Michael.
Yeah, I think I would say the same. The complexity of the genome. For my case, I've worked on cassava. For some reason, it was a bit straightforward having the reference of cassava. But this one also, like it's a new genome that has not been sequenced before, so there is not much reference to compare. And even like you go high on the family level, is then we get species that have been sequenced. So there was not much to like compare. So you actually starting from scratch. And the complexity of the level of duplication that was really sort of challenging. But of course, with the network, Sadiq also like really helped in the discussion. Thank God to within the African Assembly group. So this was really helpful. And also just to mention, like I'm really completing the last year of my PhD, so a lot going on as well. So then this was really I think a lot of work, and hopefully now we're getting to the end of it. So this is really nice. And actually sequencing, I really like the idea, thank God, like sequencing in Africa, doing it in Africa, collaborating within Africa, and building capacity within Africa. So that's really nice.
Yeah, thank you, Michael and Ansi. I think this has also been very interesting because it allows us to see firsthand what the challenges are, and also allows us to see and experience firsthand how we can actually drive and move things. I mean, looking at what you guys have done in this genome, I was telling someone the other day, they said, "Oh, thank God you called me." I said, "No, I think the genome is now on autopilot in the sense that we now need to publish it." So everything, so it's actually been very fantastic. And what awaits you guys now is how do you now take those bits, and then after it's published, take those bits and also um, uh, increase it in a geometric fashion in terms of knowledge, in terms of giving to people, in terms of, making it open, giving people the opportunities that, the experiences and sharing it, especially on the African continent, because one could sequence all this. There are institutions outside Africa that can sequence all this in a year or 2 years, but we said, no, we don't want that. We want to sequence it. And I've seen people who have also built their opportunities by just doing it because you have that sense of ownership, confidence, and things like that. So I'm really pleased with this. So there are also many of those opportunities and projects within the AfricaBP also in that same direction. And most of the time, I think to myself, what if we can scale this beyond just the number we have? What if we can take it times 10, times 20? Then that would then get us closer to the Africa we want, which all of us on this call, I believe, do want. And also that will also increase the number of researchers, for instance, because Africa has the lowest number of researchers globally. An example is that the average number of researchers per million people is around 1,400 researchers per million globally. But Africa has just around 300, 350 per million people. So which means the number of people that can actually drive things low. So we need to then increase it. And I think that is one of the significance of a project like the African BioGenome Project. And I really want to thank you, Ansi and Michael, for also showing that this can be done. And I hope that this will also provide a sense of motivation and inspiration for everyone on this call. This is a high quality assembly, high quality activities, and lots of things that experiences shared over the past few months. All right,
I think I want to just say that I think we shouldn't give up, because from what we have seen is that the project started a bit slow. Ansi, I think you also experienced that slow start as with Michael. A lot of retries, and thank you for not giving up, because eventually it worked, and that's now we're looking forward to implementing that scale globally. Yeah, thank you.
Yeah. And I think you guys didn't give up, because at some point you will say, "Oh, okay, all right, that's it, right?" So, what especially with all of this deploy polyploid, you already is just seeing the next species and doing something with it. That is where the whole thing is. All right, thank you, and Professor Ann. This was supposed to be a comment, but then it's turned into a panel session. And all right, thank you so much, everyone. Michael and Ansi, I hope you still hang around. We have a few more, one or two more presenters, and then we can wrap up for today. Do we have we have people on site? Do we still have control on site? Professor Julian, do we still have control on site to ask questions? Sorry, do we still have speakers or participants on site who want to ask questions to both Ansia and Kofia? Because I think you guys
Yeah.
Okay. Go ahead, please.
Can you hear?
Yeah, go ahead.
Yes. You have Professor Tu is here, is ready, and Dean Faculty of Agriculture is here also to give his welcome remark. And I guess we can allow him to give that remark now, so that he can go back to the meeting for where he just came back to do this now. So that after that, Professor Tu will now on the stage and make his presentation.
Perfect. That's fine. Let's do this. Thank you. Professor Julian, I think we can't hear or see the on-site participant. We can't hear the speaker as well. Is that an internet issue? Or? We lost the on-site presenters. I think we lost the on-site presenters. We'll have just one more presenter and then we will wrap up for today. I'm just trying to see if they connect back in for us to join in.
So enable your video. The slides are not working.
Okay. Hi, Professor Valentine Itui. So Professor Valentine Itui is our last speaker for today, and he's on site, but also we present via Zoom. So after his presentation today, we will then retire for tomorrow, and hopefully tomorrow we are going to start at 8 o'clock on the dot. And okay, Professor Intui is a professor at the African Genome Center in Morocco, Mohammed VI Polytechnic University in Morocco. And he's going to talk about genetic modification and its development goals in sub-Saharan Africa, application to meet the relevant Millennium Development Goals. So Professor Intui, over to you, please.
Okay. I think.
Yes, I think those on site can see, provided that the on-site network is connected. Yeah, they can see.
You remove.
So, but I think there is echo. So the on-site control will need to mute the Zoom, please. So please go ahead, Professor Intui.
Yeah, we are waiting.
Oh, I think it's also echoing. So
Hello? Hello? I should log out.
no, you don't need to log out, do you? You don't need to.
We can't hear anything. Is the presenter speaking?
Valentine, is there a chance we can just carry on so that we can finish up on time, please? So, yeah, those on site could join the Zoom. Those that have Zoom could just join with their devices so we can finish up. Yeah. All right, let's go. Let's carry on. Just carry on. You can share your screen via Zoom, please. We can't hear you. You're mute. You're mute. You're mute.
Can you hear me?
Yeah, I can hear you now. Please, let's carry on.
The presentation has disappeared.
No, it's still in your computer. Okay, all right, got it. Okay, carry on.
Okay.
the first thing the person tells you are from Africa. And when they ask anybody that is abroad, in the US, Europe, UK, which is the poorest country in the world, they will tell you Africa. Because erroneously, they think Africa is just one country. They will not know that it is a continent. And so, because there are a lot of challenges in Africa that actually has put us in a level that when somebody sees you, he sees poverty. That's what it is. When somebody sees you out there, he sees poverty. And they feel that when we go out there, we are going to beg. Yeah. But it's not everybody who goes out there goes to beg. And so, the world brought in the Sustainable Development Goals. If you look at the Sustainable Development Goals, this thing is not moving. Okay. There are 17 Sustainable Development Goals. Which of them do we actually meet in Africa? Even the most common one, no poverty, no hunger. There is none that we are meeting in Africa. What is the reason? A lot of things. Poverty, so much. You have bad governance. Even when you are not voted, you take election to go in and then destroy everything. You see, conflicts. Agriculture, that is supposed to be one of the key factors that is supposed to help us meet some of the development goals, is actually also having a problem. We don't have improved varieties. Fertilizer, we don't have money to buy fertilizer. And apparently, you will realize that agriculture actually employs about 60% of the workforce. So if that happens, then why are we poor in Africa? If it employs 60% or 65 and 70% of the workforce, why are we poor? Why are we still hungry? We should have enough food to eat, but that is not the case. And then see, it contributes about 30% of the GDP in Sub-Saharan Africa. So despite the amount of workforce that and this thing keeps on even when I don't want it to run. Okay, so then what is the problem? If that is the case. Okay, so despite that agriculture employs 65%, look at the food security. It is shown that by 2030, the food security in Sub-Saharan Africa will be up to about 364 million. That is almost 1.5 the number of people living in Nigeria by 2030. And the worst is even our giant of Africa. Look at it. It's very serious. That's Nigeria. It's a small country. And those that we fight every day with, like Ghana, they are in a very comfortable zone. But Nigeria, that is the giant of Africa, the food security situation is poor. That means by 2030, most of the people in Nigeria will go hungry. That's what it is. So with this type of situation, how do we meet the relevant Millennium Development Goals or Sustainable Development Goals? It becomes now a very big challenge. And the reason is because you have conflicts, wars, political instability, poverty. These are all man-made things. Nigeria, for example, is very rich. I am from Nigeria, Rosiba State. Very rich. When you go outside there, they ask you, where are you from? Like the first time I went to Japan, because I did my PhD in Japan, they asked me, where are you from? I said, from Nigeria. How you have money? That's the first thing they tell you. because they know that Nigeria produces a lot of oil, but that the money from oil remains in the hands of very few. And that is what is actually driving the poverty rate in this country. So that even small nations that are not supposed to talk with Nigeria, they stand up and then insult us. See, conflict, drought, diseases, high temperature, flooding, poor funding, fertilizer application that we use is not sufficient because it is very expensive. The smallholder farmers, those in the villages that are involved in agriculture, will not have money to buy fertilizer that they will apply to their farms and get high produce. And then lack of improved varieties. And so, what can make us attain the sustainable development goal could be conventional breeding for those breeders that are here. But then the issue is conventional breeding is also limiting in the sense that it takes so much time for you to develop and improve variety, so much time, okay? So one of the things that will help us increase agricultural productivity and also meet the sustainable development goal is genome editing. It might not be the silver bullet, it might not be the main key, but at the moment, it could also contribute significantly in helping us meet the sustainable development goals by actually producing plants with improved characteristics that you might not need fertilizer to cultivate. It could produce animals that are resistant to environmental challenges. It could produce plants that are resistant to environmental challenges like drought, flooding, high temperature, diseases, and so on. It could also help to boost our immunity by producing plants with high nutrient content. That could help to boost our immunity. Because one of the challenges we're having here, some of us eat a lot of starch, but the micronutrients that are needed for the body are lacking. So these are the areas that genome editing could help. Now, what is genome editing? I know that many of you have heard about genome editing, right? Yes, so what is genome editing? And as we can read together, a new tool that allows very precise and quick changes to the DNA using molecular scissors called nucleases without introducing any foreign gene. So this is a tool that you could actually configure, change the genetic makeup of an organism without even any genetic material. You just go to the organism. What is this gene that is actually disturbing this organism from performing effectively? Once the genomic people have done their sequencing and released and annotated, and then that's where we come in now and study those genes carefully. And so, oh, this is the gene that is actually disturbing this organism from actually performing their duty. And so we can go to that organism and modify the gene without affecting any other gene and without introducing any gene from anywhere. So that is exactly what is referred to as gene editing. So then in place, I think it's this thing that is. So what happens is that this genome editing requires a nuclease called scissor. So it goes to that gene, cuts the gene, and then the gene repairs itself. Okay, just like when you have a wound, that wound repairs itself. So that's what happens. So it is during that process of repairing itself that it modifies now the gene, because some genetic elements like some can be lost during that process of repairing itself. Some can added, captured from elsewhere and added maybe just one or two nucleotides, okay, resulting to a double-stranded break, and then which is similar to what happens in nature. That is exactly what happens. Like what happens in nature. What we have seen, the variety of different organisms we have seen today is because of mutation. And so the mutation that occurs through genome editing is exactly like the mutation that occurs in nature or when you do mutation breeding using chemical or mutagenesis. Okay, so in agriculture, genome editing allows the introduction of new traits and removal of undesired genes in a shorter time compared to conventional breeding. So new traits, you modify and come up with something that is acceptable. And you can see the various areas. It could enhance yield, drought resistance, clot, yield, and so on and so forth. Okay? Okay, so none of us knew about genome editing when CRISPR was developed. But before CRISPR was developed, there were tools that are involved in genome editing. And some of the tools are zinc finger replace, talent, and the almighty CRISPR. All these tools create what we call double-stranded break. So because the DNA is double-stranded, so they cut DNA because they are scissors. Okay, what the scissors does is to cut something. So it cuts the DNA and makes it. So the DNA is now in two parts in the cell. But the DNA cannot remain in two parts in the cell. It has to repair itself to be able to come back and join. Okay, so that is the process. When that DNA is trying to repair itself to come back to the same strand of DNA, nucleotides are lost. And when the nucleotides are lost, the length of the DNA shortens. And that makes it to lose its original function. Okay? If peradventure some nucleotides are substituted, the DNA sequence will remain the same, but it will still not function the way it used to be because you have reconfigured it. And if some genes or some nucleotides are added in that position that the double-stranded break occurs, the DNA molecule gets longer. And in that case, it will also lose its original function. So these are the tools that we use that are currently being used to modify the DNA in a specific place without touching any other area. It takes time to change. Okay, yeah. So because it was actually, so this is what actually made us to know about genome editing, CRISPR. So this CRISPR, cluster regularly interface short palindromic repeat, was discovered as a result of studying bacteria. So what happened was that when the scientists were studying bacteria, they realized that bacteria had some clusters of DNA that were short, but clustering it. And so they were wondering, what are these clusters of DNA that were surrounding the bacteria? So they discovered that these clusters of DNA belong to the viruses that were actually perching on the bacteria and trying to kill the bacteria. So it turned out that the bacteria was using its mechanism to cut the viral DNA so that it could protect itself from being destroyed by the virus. So it is a kind of immune system that was actually protecting the bacteria. And so that was how the scientists came up with the CRISPR mechanism that all of us are using today. So, but not
All right. Can the control side please unmute because we can't hear? Yeah.
Can you hear me? Okay, yeah. So this is one of the CRISPR tools that does not cause double-stranded base, but rather it gives just one particular base, and then it's called base editing. Next slide, if it is possible to change from there. What is happening? Go to prime editing, yeah. Go next slide. Okay. Another one that has been discovered is that you can change 2 or 3 nucleotides at the same time, and this is called prime editing. Okay. Yeah, please move, if it is possible, move. Okay, now, let me bring this a little so that we know the differences between gene editing and the traditional gene. So with gene editing, you don't isolate a gene from anywhere and insert anywhere. You should take note. Gene editing does not isolate any gene from anywhere. Rather, you modify the gene in the organism itself without introducing any new gene. Okay, but with GMO, you isolate a gene from elsewhere and then transfer to the target organism. Okay, that's one. Secondly, with gene editing, it is possible you are going to produce a final product that will not contain any foreign gene. And that is the basis by which most countries have developed a gene editing guideline. That any plant or animal product without a gene inserted in it will not be regulated as GM. And Nigeria is one of those countries that also developed a guideline on gene editing. Next slide. Next. Next. Okay, good. So let's start from here. So now we are going to see the application of gene editing in agriculture. And that is where I want us to pay attention. And this is where, if we succeed in this, we are going to actually meet some of the sustainable development goals. Okay, next slide, please. Okay, as one of the things we did when I was in IIT in Nairobi with Dr. Lina Tripathi, the head of the lab and the director of East African Hub, we established a gene editing platform for banana. So what we first did was to mutate a particular gene called the PDS, phytoin desaturase. So this gene interferes with chlorophyll. If the gene is mutated, all the plants that are coming will be white, will be albino. This is a marker that actually enables every gene person or every person that is doing gene editing to actually start with this. So once you see your plants coming out white, it means you have successfully established gene editing for that particular crop. So when we mutated, can you go back? So when we mutated the gene, all the plants that were coming were white. That shows that we have successfully established gene editing for the crop banana. So with this establishment, we went after that to see if we can produce plants with interesting characteristics. Next slide, please. So we decided to mutate the banana streak virus. Banana streak virus integrates in the genome of banana and remains in that genome throughout life cycle. And this is a problem because it will be difficult for you to transfer germplasm from one country to another if there is this virus in the system. It behaves similar like the AIDS virus because the AIDS virus infects and then gets integrated into the system. And so one of the things we did was, could we inactivate that virus so that it will not actually create symptoms? So when the virus integrates, it has two forms. One is that integrated form, and another one is the episomal form. So when it gets integrated, it is dormant. But when conditions become unfavorable, so that integrated version reverts to episomal, the round one, this is the episomal version, and start creating the symptoms. Okay, so what we did was, next slide, please. what we did was we inactivated that virus so that when conditions are unfavorable, it will not revert to the episomal version and it will not also create symptoms and the disease. And so as you can see, most of the plants here that we mutated were free of the symptoms, meaning that we successfully mutated the virus and inactivated it in the banana system. Next slide. So another thing we did was, could we produce banana for resistance to a particular bacterial disease? In East Africa, there's this disease that is attacking banana, they call banana xanthomonas weevil. It is very serious in East Africa. And so one of the things my boss, Dr. Lena, said, could we go on to actually produce banana that will be resistant so that the East African community can benefit from banana that is resistant to that particular disease? We inactivated, we mutated this gene called DMR6 in banana. And the result was that, next slide. And the result was, if you look at this disease-resistant assay, on the right-hand side is the control, the one that the gene has not been mutated. On the left-hand side is the mutant, the one that we mutated. When they were subjected to, when they were challenged with the disease, you see that our plants that had the mutation for that particular gene were actually strong. They did not produce any symptoms. Okay, so that means if you mutate that DMR6 in banana or any other plant, you can possibly produce resistance to bacterial disease, as we have seen in the case of banana. Next slide. Another gene we targeted, because we were trying to experiment a lot of genes that were already dead. Another gene we targeted was early nodulin. Next slide. So again, when we mutated this gene and knocked it out in banana, see what the result that we got. The lower bottom right is the wild type, WT. That means the one that is not mutated, does not have mutation in that gene. And the other ones are the ones that have mutations. Okay, you could see that the wild type plants died completely after being challenged with the bacteria, whereas the one that mutated the gene had no symptoms. That shows that this particular gene too is very effective. And so that is why, see, these genomic things we do, we have to study the genes very well. Once the bioinformatician or those that have sequenced the gene, sequence and notate the gene, now it is our duty for us to go into those genes to find out which particular gene is based on the annotation for which target. So we work hand in gloves with this gene. Next slide, please. Next slide, I want to skip this one. Okay, now this is another work. This work was not done by us. This one is done in tomato for resistance to the virus. Okay, so they mutated the gene that is causing resistance to this virus and powdery mildew. So in the end, what happened? They got plants that were resistant to this particular virus. Now you see how gene editing is working. I'm moving now from one plant to the other. Next slide. Now, this issue of hidden hunger that we experience in sub-Saharan Africa, whereby we eat a lot of eba, no protein in the soup, no vitamins, no zinc, no iron. You see somebody tomorrow, he has kwashiorkor. Another, because what we do is we eat to fill the stomach. We don't eat to say whether we have a balanced diet. But then, because we depend so much on the food we cultivate, like yam, cassava, rice, and so on, and because we are poor in sub-Saharan Africa, we don't have the money to buy a lot of things, so we eat what we see. We are not able to supplement the diet, and so balanced diet becomes a problem. But then with gene editing, even if you don't have money, you can still have nutrients from plants. So what these authors did in banana was to increase beta-carotene, the precursor of vitamin A, You know, lack of vitamin A leads to blindness. You know that, right? So they increase the level of vitamin A through beta carotene. So they mutated the gene that this gene here, the one that has a cycle, lycopene epsilon cyclase. When that gene was mutated, they increased provitamin A in banana. You could see the yellow one has high amount of provitamin A compared to the control. So that means through gene editing, you can create disease resistance and you can actually reduce hidden hunger by increasing the level of vitamins in the plant. Next slide. Yeah, this is another work done by another group. The first author of this paper is presently working with me in UM60 as a postdoc. So they increased the level of iron and zinc in wheat. Iron and zinc in wheat by mutating this gene here in that is the blue area, IPK1. When that gene was mutated, the level of iron and zinc compared to the controls, if you can see on ABC, was highly increased. And iron and zinc is one of the things we are lacking in our system, especially in Southern Africa. And if we can make plans to accumulate these micro elements that are very essential, then we'll be able to solve the issue of hidden hunger. And that would lead us to meeting up with Sustainable Development Goal 2. Next slide, please. Another thing that another group did, could we actually reduce the size of a particular plant? You know, when plants are very tall and there is wind, it can blow off. Yes, the topple, especially something like banana. But if we can make short plants that will be able to, even if you stand, you can harvest. That would be a very good thing for us. Okay? So that even when there is wind, those plants will not topple, but rather they will strengthen and they will be able to produce. And that will lead to increasing productivity. And so what they did was they interfered with the gene MAGA20 in the gilberellin pathway and produced plants that were comparatively shorter than the wild type plants, as you could see from that picture. Okay, next slide. So another thing we are facing in South Africa, that's why we cannot meet these sustainable development goals, is lack of storage facilities. In Southern Africa, the weather is too hot. You harvest something today, by next day it decays, especially in Nigeria, we don't even have electricity. Yeah, and the market, ready market is not there, right? So if you harvest your tomato and keep it in the house, by tomorrow it will decay. So what gene editing did, like these people, could we increase the shelf life of plants so that when we harvest our fruits, they can stay for long before even they decay. So that by the time they start decaying, we would have seen the market. People would have bought. So, but they demonstrated this in banana by editing this gene, MAC01 is a very long name. Aminocyclopropane 1 carboxylate oxidase 1. When they mutated that gene, look at the banana. The white type started decaying after 21 days. And the one that had mutation in the plant stayed up to 80 days, almost 2 months. So if you are a tomato farmer and we produce tomato by mutating the gene and give to you, you say hallelujah, because we'll be able to harvest your tomato and store them for long, even if you don't have storage facilities, because no electricity. I cannot count the number of times light went out here as we're in this hall. And what of now, the smallholder farmers, now they don't have money. You see? So that is another way that gene editing can contribute. Next slide. Nitrogen use efficiency. You know, we apply a lot of fertilizer to our plant, and nitrogen is one of the most required nutrients, right? But at times, the plants don't absorb nitrogen, and as such, they will not be able to produce effectively. And so, by modulating this gene, AR1, you could make the plant to absorb more nutrients, nitrogen from the soil, okay, and use it effectively and develop well. And that's one of the key areas now of research, how we can make plants to absorb more nitrogen and phosphorus from the soil. Okay, so that even if it is a little fertilizer that is applied, the required amount of nitrogen that is available in the soil will be absorbed to make the plant more effective. So when they mutated this gene, you could see the white type plant is on the left. Okay. The other ones, even the leaves, see how green they are. That means they have used that nitrogen effectively. And this is one of the things we could think of doing. Instead of buying a lot of fertilizer to apply when you don't have money, if we can get these seeds and cultivate, you'll be able to reduce the amount of money. You can keep that money for other things. Next slide, please. Drought tolerance. A lot of things, especially like in East Africa, drought, environmental changes, okay? Because environmental change is having a lot of effect, drought and so on. Now, they mutated the gene Wiki5 in rice, and were able to produce plants that were tolerant to drought compared to the wild type. I'm bringing this thing from different areas to see how gene editing is more effective in achieving this type of result. So if you could see, these are the white type plants on the right in those yellow boxes. You see how they already died. But when they were subjected to drought, look at gene will already die. Next slide. For those of you that are interested in mushroom farming, it is a mushroom and key. What happens to it? If it's left for long, it becomes right. Yeah, but gene editing was able to produce mushroom that can stay white all through. And it was commercialized in the US some time ago. That's it. All of them are white. You see? So that means it reduced that browning activity because browning leads to what? To decay. Next slide. This is one of my best slides. Now you check we have 3 slides there for animal. I was showing all through plant, now it is the case of animal. In the first case, the cattle. What did you notice there? Have you noticed anything in the cattle? No horns. Yes? Gene editing produces cattle without horns. Why? Why should I bother producing cattle without horns? Their aim is that when they are together, they fight and they kill each other. I would not want to lose my cattle. And so they were able to produce gene-edited cattle without horns by mutating that gene. Pollet allele, easy, and they achieved that successfully. Now, chicken, bird flu always attack our chicken, especially for those that are doing poultry. Within a space of no time, you see that your chicken is gone. And so they produce the chicken for resistant to this disease, bird flu. But the most important is tilapia. Imagine producing red tilapia. That was done in Israel. And the tilapia was no longer sellable. People did not like it that much. And so what could they do to make sure that our market continues and remains relevant in the field so that we'll be able to children and send them to school, that gene over there. Next slide, please. Hello? Okay. Your thing has scattered. And so this thing really good. So this is one of the most interesting aspects of this gene editing. You know, at a particular age or based on the activity, you start having high blood pressure, all right? And once you start having high blood pressure, a lot of things, you start controlling your diet and so on. You start buying medicine.
Professor Ntue, do you want to unmute yourself? We can no longer hear. Yes, unmute that one because we lost video. Aha, all right, that's fine. All right, perfect.
Can you hear me?
Yeah, we can hear you now. Carry on.
Okay. Hello, can you hear me?
Yeah, we can hear you now. Carry on, please.
Okay, that's fine. So one of the things of this application of genome editing is in medicine is sickle cell. So what they did was they mutated CD34 cells. Okay, the CD34 cells that are part of the cell of BC11A, a transcription factor that suppresses gamma globulin and hemoglobin, because those are one of the things, those are the things that affect sickle cell. And so when they did that and administered to 2 patients, they all produced increased level of hemoglobin. You see? So that means with time, genome editing will be able to treat sickle cell. So AS and SF will no longer have issues. They can be married now. Yes, with time, because some of the things is already happening. So you see, low blood pressure, sickle cell is already, so research is still going on. Next slide, please. Next slide, please. Okay, I'm done now with research. Now I'll show you the regulatory, because you know, at times when something comes, new research comes, they will try to make regulations and so on, so that to regulate the way research is done and how the acceptability and so on. One of the things is, fortunately, Nigeria actually was the first to develop the guidelines for gene editing in Africa, then Kenya, and then now we have Ghana and Malawi. So that means we could do the research and take the product to the field. And in Nigeria, even though it's a case-by-case basis, any product that you produce without a foreign gene integrated or a foreign element is considered as what? Non-GM. You just take it to the field. It's just considered as conventional breeding. Anyone that is just grown by conventional breeding or mutagenesis. And that is the advantage we have with some of the African countries. So with this type of thing, then we should increase research in genome editing. Next slide, please. These are some of the projects that are already going on in sub-Saharan Africa. And a couple of them are in Nigeria. A couple of them are in Nigeria that are going on gradually. With time, there will be a boom in gene editing product in this country. And I think it will help us to solve the issue of agriculture and help meet the relevant sustainable development goal, which I will show how CRISPR and which one can CRISPR or gene editing contribute to. Next slide. So the African Orphan Crop Consortium is training African scientists on genome editing. So far, they have already trained 21, and presently there is a third cohort that is undergoing the training in Nairobi. When I was with IITA in 2023, I was also part of the group that was training. You could see from this number, of course, the Giant of Africa has 6 already trained. And some of them are from NAFDA, University of Calabar. There was nobody from Unicorn, unfortunately. I don't know whether this year anybody has applied. So for those of you that are scientists that are interested in doing genome editing, especially for those from agriculture and plant breeding, you should look for this and apply. So the first cohort that graduated in 2023, they were given some seed funding of 15,000 to establish gene editing platform in the country. And from time to time, I go visit them to check what they are actually doing, to encourage them so that we'll be able to actually develop those skills in the different African countries. Next slide. Now, this is where gene editing, based on all the talk I've given, the application in agriculture, in medicine, in animal science, in plant science, and so on. These are the areas that gene editing can contribute. One, no poverty. If gene editing can help us to reduce plants and animals that are resistant to environmental challenges. And we have seen that from the beginning, I said that agriculture employs about 65% of the population of sub-Saharan Africa. But then the output is very little. And so that's why we still have hunger, we still have poverty, and so on. So if gene editing can help us produce plants and animals that are resistant to environmental challenges, then we are going to meet some of these. No poverty, no hunger, good health, because of micronutrients deficiency that we have in hidden hunger. And if you have money as a result of your agriculture, you will be able to train your children in school very well. Quality education. Clean water, reduce amount of fertilizer because of your having plants that are already resistant to a lot of things. You will not be able to apply fertilizer, then there will be no leakage to water to pollute the water body. Even the fish in the ocean you have seen. So these are the areas that gene editing can actually contribute. What we need to do is to embrace it, to give sponsorship to it, so that we could stand. But I'm not saying that it is the only thing. It's one of those things that we could use to actually meet the relevant development goals in sub-Saharan Africa. So that whenever we go out there and somebody sees us, they will not see me and see poverty. Exactly, because that's what it is. When they see us, they see poverty. Of course, our people will not keep drowning in the Mediterranean Sea trying to cross the sea to Europe. All what we need is, we need some of these things, but then we also need the government support. Yes, if government in the different countries, and that is why I think at times we do, and we talk also politics. Ibrahim Traore will support that young man. If all our leaders can stand up like that man, I think we will make a big change in this, in Sub-Saharan Africa. Next slide, please. So the take-home message here is that, I'm ending, genome editing is an important tool which will be adopted to increase agricultural productivity to meet the SDG goals 1, 2, 3, 4, a lot of them. Genome editing can produce crops and animals without any foreign gene incorporated into the genome, hence will not be regulated as GMO. Next slide, please. Sub-Saharan Africa is encouraged to adopt GE tools to increase agricultural productivity in the region. And few SSA countries have developed tools. So we expect others to develop the guidelines so that editing can occur in the country. Okay. And of course, increased training in some SSA students and scientists to facilitate the adoption. And that's one of the things African and Open Crop Consortium is doing by increasing training. This year, I joined 160 last year. We have four PhDs, one from Nigeria, from University of Calabar, two from Morocco, and one from Ethiopia. Usually we advertise every year. I also have two postdocs training them on gene editing, one from Nigeria from the University of Calabar, and one from Pakistan. So if these students are trained, they will go back to their country and start to develop gene editing. Next slide, please. I'm almost getting the next slide, please. Hello? Okay, please go back to the former slide. Okay, next slide, let me see. Yeah, so this is now my university. University Mohammed VI Polytechnic in Morocco. To be honest with you, I have been to several countries, as I said, PhD in Japan, conferences in Europe, in America, postdoc in Saudi Arabia, King Abdullah University of Science and Technology, IITA.
Okay. Thank you very much, Professor Ntui, for the very nice presentation. So I don't know if anyone have any further question? Okay. I think he's still. It looks like he still has more slide. Let's see if he's finished up already. Professor Ntui? Yeah, I think we can no longer hear you. So could you please maybe we can wrap up and maybe take questions because it looks like we're going past the schedule of today's proceedings. Can you please unmute? Professor Valentine, could you please unmute? We can't hear you. The other video is
Okay.
Yeah, okay. So we have some speakers so that we can quickly wrap up for today. We have some persons who would like to ask some questions. Is that okay from this end, from the virtual participants? You're on mute again. You're on mute.
Okay.
Okay, yeah. Stephen, could you please ask your questions?
Oh, okay. Thank you very much. I want to thank Professor Ntu for that beautiful presentation, the vigor, and I particularly like the introduction where he kept on emphasizing on the issue of food security and then, of course, the sustainable development goals. My question is, in fact, the three presentations this afternoon were biased on the crop, but eventually Professor Ntu talked about animals, livestock. So For livestock, he gave one example of the benefits of gene editing as for producing animals without horn or hornless animals. I would want to know if there are other benefits apart from producing hornless animals. And again, he made mention of the benefit in humans and the prevention or treatment of sickle cell. I would want to ask if, of course, we have such opportunities in preventing livestock diseases, I mean animal health. Thank you very much.
Professor Ntu, over to you, please. You might need to come close to your laptop to answer the question because the other camera, we can't see it. So
Can you hear me?
Yeah, could you please stay close to your laptop to answer the questions because we lost the other camera in the.
Okay, can you hear me?
Yeah, we can hear you. Go ahead, please.
Okay, yeah, thank you for that question. Yeah, you know, when the researcher that is the first benefit. So but that is not the only thing in animal do. So it depends on actually what you want in question. And that is what you are actually going to do to benefit you, because each thing that you do, you have a particular target. That's one. So in the case of medicine, you know, it's just coming up. Gene editing is new, let me say so, because it's just about the past 10 years. But a major development was done in plants because it is easy to manipulate plants than animals. And then again, in animals, a lot of ethical approval is required for you to work with animals. And so because of that, that's why we're seeing a lot of work done on plants and gradually moving from plants to animals and then to humans and medicine. So maybe in the next future, we are going to see a lot of studies that will be released, because I know that people are already studying how they can cure HIV using gene editing, like the system we showed for banana by inactivating the banana streak virus. So work is already going on how they could inactivate the HIV virus. And I think with time, we are going to have something like that. Most probably also for cancer, things are already going, but I've just selected these few that tests have already been done. So I think with time, we are going to see robust in all these areas.
Thank you, Professor Ntu. So we have another participant who would like to ask a question. Linus, could you please ask your question?
Cannot hear. Let me increase my. Sorry, let me increase my my mic.
Linus, could you please ask your question, please?
Yes.
Okay. Thank you so much. I'm very pleased to listen to my teacher speak. Professor Ntui is my teacher, right? Right from BSc and through my PhD. Prof, it's a great pleasure listening to you again from far away. Yes, you know where I am. So, Prof, this is a quick one. Have there been successful trials, because you've done a lot of research in this area? Have there been successful field trials? Because the end success of every research is to have it, as a byproduct, as a product that is consumed by the end users. Have there been successful trials of these discoveries that you've made, especially in the area of development of resistant varieties, like you spoke in banana? You also spoke about the knockout of some gene, and also the development of short species of these varieties. That's one of the questions. If there have been, where and where? Because it's easier to have an in vitro experiment where we can say we've achieved success in the lab, but is it successful in the field? That's my question. Then the number two question, I would love to find out your school, the UM6 Polytechnic, you spoke about a lot of opportunities for postdoc because your area is is germane. So how possible can one achieve that? So are there a particular period where you advertise for postdoc students and all that? Thank you.
Thank you, Dr. Linus. I know you are in Malaysia. So
thank you for that question.
So
are going on. Which one? I should mute mine.
Yeah.
Can you hear me?
Yes, please go ahead.
Yeah. So for banana that we developed, field trials have not yet started, but so far the greenhouse experiment, not in vitro, has shown a good result. And so my boss, Dr. Lina, who actually is the head of this, is planning on doing field trial with some of the varieties. But for tomato, that has already been commercialized. Field trials were done, and that's why they commercialized that tomato. Okay, so I think I've answered it. And then for opportunities, you know, it depends on the area. There are a lot of areas in UM6P, and each day they advertise positions ranging from postdoc to faculty positions. Yeah, so if you put an eye on success factor, UM6P success factor, you'll be able to find out. Because if you open it presently, you see about 150 positions. And for PhD, I think from by the end of May or early June, you are going to have a lot of PhD positions that will be open. So it's just for you to keep an eye on that in that area. Then you'll be able to see which position suits you, and you can apply. But when applying, take note, you are not the only one. Because are you applying other people are also applying. At times there could be 150 applying for a particular position. So you have to assemble your CV very well to actually convince the professor who actually posted the postdoc or whatever thing.
Thank you, Professor Ntue.
Yeah, I've muted on Zoom. No, I can mute the main system.
No, it's fine. The main system is muted. So, do we have any more questions for Professor Ntue? Any question on? Any question
on site? There's a question.
All right, please go ahead on site.
Yeah, my question, Professor Tobias Innocent, Indonesia, from Federal University of Technology. My question is similar to what I've been asked, because well, in the context of the program, we're talking about genome in One Health, and we know that One Health brings in animal, brings in the environment, brings in human. And so, on the context of your teaching, one will want to see you explain that of plants, banana, tomato, and all whatnot. And we know that environmental issues is a very big driver to health issues. Degradations. We know that organisms are key players in degradations, which help to mop up problems from the environment. And so what we want is to have an overall success in One Health, as it were. Thank you.
Yes, that's a good. Yeah, that's a good question. Yes, there's not nothing we cannot do in genetics. But you see, you can use plants. You can modify plants to actually capture heavy metals from the soil by targeting a particular genes. Okay, I submitted a proposal to OCP to modify plants to capture heavy metals by targeting some genes which I will not mention here. So that's one of the things we can do. You can also modify bacteria through gene editing to capture heavy metals from the soil. And one of the things that our center is trying to do is to modify bacteria to capture cadmium, remove cadmium from the soil, so that soil could, soil health could be achieved. So gene editing can do a lot of these things. It depends on actually what you want to do and how you are looking at it. So there are a couple of genes that once once we know the genome of several plants and the genes are mutated, we'll be able to actually check which genes we can modify that will be related to capturing some of these things. And so projects are going on in several areas. I just pick a few I've mentioned here because I cannot mention everything. I've mentioned animals here. I've mentioned also human health. Yeah. So that is it. Yeah. With that, you will see some of these things being done.
Yeah.
Okay.
Thank you, Professor Ntwe. I think there's one more question on site, and then we can come back to Zoom.
Thank you. Prof, thank you for that presentation. My name is Dr. Lawrence Okonko. You have presented genome editing as a very beautiful bride, and personally, it has reaffirmed my love for that technology. In the course of the lecture, you distinguished between GMO and crops that are developed using genome editing. Are there safety concerns? Because I know that GMOs have safety concerns. And as an ordinary person, ordinary Nigerians on the street, they're always conscious of their safety, especially as it has to do with what they consume. What are the safety concerns associated with genome editing as it concerns crops? And what are the measures put in place to check this? And then finally, is there any danger as it concerns the interaction between these crops that are developed using genome editing and the wild type? Thank you.
Yeah, thank you. You see, some of the things that people say about GMO, about safety, about that, are things that people just say without actually going into the detailed research about it. Before anything is released into the environment, there is a lot of tests. A lot of tests has been done, a lot of screening, a lot of both genetic screening, physical screening, and so on has been done before being released. And so for you to see something being released, it means it has passed a certain threshold that there is no safety concern. I know people always talk of horizontal gene transfer. That's one of the things that they usually say. But up to date, nobody has actually proven that there is actually horizontal gene transfer using GMO. Now that's on the one side. And then I said about gene editing. You see, when you modify an internal plant, a gene that is endogenous in the plant, just like when you do chemical mutagenesis. If you do chemical mutagenesis by subjecting the plant, okay, to chemical that will actually create mutation in the plant, and you take that to the field, do you have any concern about that? About safety concern? And what would be the safety concern?
Concern should be the fact that the method, sorry, the chemical with which that was created is likely going to pollute the environment. I'm going to pollute every other organism outside the
But that chemical mutagenesis is done in the lab, you are not doing it in the field. So what is talking about field? Once you mutate a plant and take it to the field, now those safety concerns are not there because you are mutating endogenous gene. You are not introducing any foreign gene. So far, and to the best of my knowledge, all the plants that have been produced either through genetic modification, that is a GMO, or through gene editing, have considered to be safe. And the one that actually go to the field very fast is the one even produced by genome, because the Nigerian regulation says if you modify a plant through genome editing and screen and show that there is no foreign element, genetic element integrated in it, it is considered as natural mutation. And if it is considered a natural mutation, then therefore there is no safety concern. That's it. Okay, thank you. Hello. It's from Desmond.
Okay,
Hello.
thank you, Professor Ntui. So I think we have 5 minutes, within the hour. So Desmond, could you please very quick with your question so we can wrap up?
Okay, good day, Prof. Ntui. My name is Desmond Chineke. I'm one of your students from the University of Calabar, and it's always a pleasure listening to you speak, especially as I particularly have growing interest in the field of genetic engineering. So as regards CRISPR-Cas9 genetic engineering, while we look at all the flowers and all the good parts of it, there are particular concerns, especially for someone like me who have heard of off-target. So I want to know, we would like to know how accurate and specific CRISPR is in targeting just the intended gene for editing, and if there are potential long-term health consequences of unintended mutations that may arise later on. Thank you very much.
Yeah, thank you. That is even what I thought you were going to ask, the off-target mutation. Yeah, so there's a possibility that you might have off-target mutation in gene editing. That is, that's why you don't want to go to do scientific. The gene that you intend to edit, you edit that gene, and again you edit another gene. Okay? So when you edit your own gene and edit another gene, you are going to produce something that probably that is not what you wanted to produce, right? And that is a concern. That is a problem. But then, before we even go to producing, editing a gene, there are a lot of bioinformatics we do. First, when we design our guide, because that is actually what guides the Cas9 to a particular position to edit the gene. So there are tools these days that have been developed to actually tell you whether your guide is going to edit another gene or not. So when we design our guide, we select a guide that has edit only the target gene without touching any other gene, because we don't want to touch any other gene, and that is where the precision comes in, precise and targeted mutation. So for the scientist that does the work, select a guide that will only edit the gene of interest without editing any other gene. By so doing, you will avoid off-target mutation. But if you are a blind person that is just going into the field and you design a guide without going through to check whether that guide will also edit another gene, then you might end up editing several genes with that particular guide. And that is the problem. So most of the crop that we produce, we only edit the gene of interest because the guide that we select will edit that gene and only that gene alone without editing any other gene. So this is very tricky. You must have to know a lot of bioinformatics when you are selecting your guide, and also know the genome of that plant very well, so that even when you edit your guide, you also blast it against the genome of the plant to check whether that guide is hitting any other plant, any other gene. If it is hitting any other gene, then you have to be careful not to select that guide. The challenge now becomes when the plant does not have a genome sequence. That's where the plant becomes. And that, I think, is what the African BioGenome Project is trying to do, to help us sequence all the African-based plants and animals so that we'll be able to know their genome sequences. And when we are doing genome editing and we design our guide, we always select the guide that will mutate the gene of interest.
Thank you, Professor Ntui. I think if we leave this conversation, it can go on and on. So I think, Professor Ntui, you could drop your email in the chat box, and then the participant could actually reach out to you.
Thank you.
So, yeah, so with this, I think we would I'll hand this over to Professor Osoje to give the vote of thanks and close the session. Prof, I think we're at the top of the hour. Could you please give the vote of thanks, and then we wrap up this session? Thank you.
Yeah, thanks so much, Tango. And actually, our program was supposed to start at 8 o'clock, and we have the logistic issues at the beginning, and somehow we're able to come on board again, and it has been just great. I know many of you who are here have come from far. For instance, the first person that actually arrived here that called me came from Kano. Yeah. Please, could you rise up, please? Let's give him a round of applause. We came from Kano, and the first person to get here. Yeah. They usually come out as a team, but for some time now, because of council coming and a lot of things, they have been very busy. So you can say today, he managed to just sneak out to come and spend some time with us. So we are actually grateful to the Vice Chancellor for this doing the needful, and there are so many people here who are lecturers in other Nigerian universities. We all know what it is like in Nigeria now with lecturers. And so those who were able to come, despite those challenges, I think we really thank you for their effort to be here. And the students who have come from different places. For the University of Port Harcourt, the students are actually on holiday. The University of Port Harcourt will be open to studies on 18th of May. And many students had to leave their homes, had to leave very badly, spending that day to come here to attend this event. And some students also from other places who came, all us and got there. Please, we are very happy that you came, and I am sure you have value for your money and time. So thank you for coming. Ambos, who is also there,
Prof, we'll still meet tomorrow. Prof, we will still meet tomorrow. We have the day 2 tomorrow. So maybe if we don't want you to exhaust the vote of thanks for us today, so that we can have some for tomorrow. So network. Thank you, everyone. So I think he's just thanking all the speakers for today and the persons who attended. Tomorrow we will start at 8 promptly. We won't be spending the kind of starting late as we did due to the unforeseen technical challenges. So today, well, tomorrow we'll start early. So please, at 8 a.m., please join the workshop. With this, I would like to thank every one of you for joining, and I will see you all tomorrow.